Hello there, and welcome to Sleepy Science Vault. There is something quietly extraordinary happening inside your head right now. As you settle in, as your breathing slows and the world outside begins to feel a little further away, your brain is doing something it has been doing every single moment of your life. It is holding on to things. Impressions. Feelings. The sound of a voice you haven't heard in years. The smell of somewhere you used to go as a child. The particular weight of an afternoon you can't quite name but somehow still carry with you. We call all of this memory. And for as long as human beings have been curious about themselves, memory has been one of the deepest mysteries we've tried to understand. Tonight, we're going to begin exploring that mystery together. Slowly. Gently. There's no rush. We're going to let these ideas drift through your mind the way that light moves through still water, without force, without urgency. Just a quiet kind of wondering. Let's begin. The storage capacity of long-term human memory is, for all practical purposes, immeasurably large. When people first start thinking about memory, they often imagine it like a hard drive, a finite container that can eventually get full. You've stored too many things, and so something old gets pushed out to make room for something new. It's an intuitive idea. But it turns out to be almost entirely wrong. Neuroscientists who have tried to estimate the raw storage capacity of the human brain have arrived at numbers that are genuinely difficult to hold in your mind. Some estimates suggest the brain can hold somewhere in the region of 2.5 petabytes of information. A petabyte is 1 million gigabytes. To put that in some kind of context, if your brain were a digital video recorder, that capacity would allow you to leave it recording continuously for roughly 3 million hours before it ran out of space. That's more than 300 years of unbroken footage. And even that number may be a significant underestimate. The truth is, we don't fully know how to measure it. Memory doesn't work like digital storage, where each piece of information takes up a neat and calculable amount of space. It's far stranger and far more interesting than that. But what we can say with confidence is this: you are not running out of room. Whatever you have lived, whatever you have felt, whatever you have learned, there is space for all of it, and then vastly, almost incomprehensibly, more. Let that settle for a moment. The space inside you is enormous. The brain does not store memories in one central location. Memory is distributed across the entire brain. It's a very human instinct to look for the place where a thing lives. If you lose your keys, there is a specific spot where the keys eventually turn up. If you want to find a document, there is a folder it belongs in. We naturally carry this logic into the brain. We assume that if we could just find the right spot, the right region, the right structure, we would find the place where memories are kept. But the brain doesn't work that way. There is no memory vault. There is no archive room tucked behind your left ear. Memory, it turns out, is not a location, it's a pattern. And patterns, almost by definition, don't live in one place. When you remember something, when a particular memory surfaces, what's actually happening is that many different regions of your brain are becoming active at the same time, each one contributing a different thread of the experience, and together, they weave something that feels like a single, unified recollection. The color of the sky on a particular day might be held in one part of your brain. The sound of what someone said might be in another. The feeling in your chest when it happened, somewhere else again. Memory is more like music than it is like a filing cabinet. A piece of music doesn't live in one note. It lives in the relationship between all the notes, in the space between them, in the way they move together. Memory is the same. Memories are not stored in individual cells. They are stored in the connections between cells, in neural circuits. If you took a single neuron out of a brain and looked at it very carefully, you would not find a memory inside it. Not even the ghost of one. A single cell, in isolation, holds nothing that we would recognize as a recollection. What holds the memory is the relationship between cells. The way one neuron has learned to reach toward another. The particular strength of the signal that passes between them. The intricate web of connections, called synapses, that links thousands or even millions of cells into a pattern that your brain can reactivate, like striking a match to light a fire that knows exactly which shape to burn in. These webs of connected cells are called neural circuits. And a memory, in the most physical and literal sense we can currently describe, is a circuit. When you remember something, you are rerunning a pattern of electrical activity through a network of cells that learn to fire in that particular configuration because of something that once happened to you. It's a remarkable thing to contemplate. Every memory you have ever formed has, in some very real sense, physically changed the architecture of your brain. You are, in a way that is more than metaphorical, made of your experiences. The specific cells that store a memory are called engram cells. Scientists have a word for the cells that are recruited into a memory circuit, engram cells. The word engram itself is older than modern neuroscience. It was proposed over a century ago by a German scientist named Richard Seemann, who imagined it as a kind of physical trace left behind by an experience, the way a stylus leaves a groove in wax. For most of that century, the engram was a theoretical idea. Scientists believed it must exist, they could reason their way toward it, but they couldn't find it. They couldn't point to a cell and say, "This one. This cell is part of the memory of that moment." In recent decades, with the development of extraordinary new tools, neuroscientists have finally been able to identify engram cells directly. They can tag the cells that become active during an experience, observe how those same cells are reactivated when a memory is recalled, and even, in remarkable experiments, artificially stimulate those cells to trigger the memory from the outside. The engram is no longer just a theory. It is a measurable, observable, physical thing. And somewhere inside your brain right now, there are clusters of cells quietly holding the shape of everything you have ever known. Memory is stored with built-in redundancy, meaning the same information is encoded across multiple overlapping circuits, not just one. Here is something that might feel counterintuitive at first, but is actually one of the most elegant features of biological memory. The brain doesn't just store a memory once. It stores it many times, spread across overlapping networks, so that even if some of those connections are damaged or lost, the memory can often still be recovered. This is what engineers call redundancy. When something critical needs to be protected, you don't keep just one copy, you keep several, distributed across different locations, so that no single point of failure can destroy the whole thing. The brain, shaped over millions of years of evolution, arrived at the same solution. This is part of why people can lose portions of their brain to injury or disease and still retain vast amounts of their memories. It's part of why memory is so surprisingly robust, so resistant to being erased cleanly, all at once. Memories don't tend to vanish like a file being deleted. They tend to fade gradually, or become harder to access, because the brain is working with a damaged copy of a distributed pattern rather than losing the only copy it had. The brain protects what matters, and it does so quietly, automatically, in a way it took us a very long time to understand. Different aspects of the same memory are stored in different regions of the brain. When you remember a particular experience, let's say a meal at a place you loved, with someone you cared about, what you experience as a single unified memory is actually assembled from parts that live in very different neighborhoods of your brain. The visual details, the look of the room, the color of the light, are tied to your visual cortex. The sounds, music playing, the murmur of other people, the particular way a laugh sounded, are stored in regions associated with auditory processing. The emotional texture of the memory, the warmth of it, the slight wistfulness, that involves structures deep in the brain, particularly the amygdala, which is closely involved in processing feeling. And the sense of where and when it happened, the spatial and temporal context of the memory, draws heavily on the hippocampus. All of these pieces are stored in their respective home regions, and when you remember, they are drawn together into something that feels complete and whole. The brain is the world's most sophisticated assembler of experience. It takes what was once scattered and makes it, for a moment, feel perfectly unified. Different types of memory use completely different brain regions. Not all memories are the same kind of thing. And the brain, which is exquisitely specialized, handles different kinds of memory with different biological machinery. There is declarative memory, the kind of memory you can consciously access and describe. The name of a city, what you had for breakfast, the year something happened. And within declarative memory, there are further distinctions: episodic memory, which holds your personal experiences, the events of your life as you live them, and semantic memory, which holds facts and knowledge about the world that have become detached from any specific moment of learning. And then there is non-declarative memory, the memories you carry in your body rather than your mind. The way your hands know how to tie a shoelace without your having to think about it. The physical skill of riding a bicycle. The conditioned response that makes you flinch at a sudden loud noise. These kinds of memory don't live in the same places as your conscious recollections. They are stored elsewhere, processed elsewhere, retrieved by entirely different mechanisms. The brain is not one memory system. It is many memory systems, running simultaneously, each one handling a different dimension of what it means to remember. Different types of memory live in entirely different neural circuits. Following naturally from what we just considered, because different types of memory use different brain regions, they also run through entirely different neural circuits. The pathway that forms a childhood recollection is not the same pathway that teaches your fingers how to play a chord on a guitar. Declarative memories, the ones you can speak aloud, rely heavily on a circuit that involves the hippocampus and the prefrontal cortex. Procedural memories, the kind stored in physical skills and habits, are processed primarily through a circuit involving the basal ganglia and the cerebellum. Emotional memories draw heavily on the amygdala. Conditioned responses involve the cerebellum as well as certain brainstem structures. These circuits don't just differ in the brain regions they touch, they differ in how they form, how long they take to consolidate, how vulnerable they are to disruption, and how they are ultimately expressed. They are, in many ways, running different software on different hardware, even though they all share the same skull. This is part of why it is possible to lose certain kinds of memory while others remain perfectly intact. A person with severe damage to the hippocampus may be unable to form new conscious memories, and yet can still learn a new physical skill, improving day after day, even while having no memory of ever having practiced it. The circuits are separate. They fail separately. And sometimes, in those failures, we see most clearly just how many different things we mean when we use that single word, memory. Memory follows a three-stage life cycle: encoding, consolidation, and retrieval. Every memory you have ever formed has passed through three distinct phases, each one necessary for the memory to exist at all. The first stage is encoding, the moment when an experience is first taken in and translated into the biological language of neural activity. Not everything that happens to you gets encoded. Your brain is constantly filtering, prioritizing, deciding what is worth registering and what can be allowed to pass without a trace. Attention, emotion, and novelty all influence what gets through. The second stage is consolidation, the process by which a freshly encoded memory is stabilized and strengthened over time. A new memory is initially fragile. Like something written in wet ink, it can be smeared or lost entirely if it isn't allowed to dry properly. Consolidation is the drying process. It happens partly while you are awake, but importantly, and we will return to this many times as we go further, it happens most powerfully while you sleep. The third stage is retrieval, the act of calling a memory back. And retrieval is not as passive as it might sound. Remembering something is not like pressing play on a recording. It is an active reconstruction. The brain reassembles the memory from its distributed pieces, and in doing so, it is always, subtly, rewriting it. 3 stages: encoding, consolidation, retrieval. Every single memory you have ever had has made this journey. The hippocampus acts as the brain's memory solidifier. It takes new experiences and gradually integrates them into long-term storage. If there is one brain structure that appears again and again in the story of memory, it is the hippocampus. Tucked deep within the medial temporal lobe, one on each side of the brain, shaped somewhat like a seahorse, which is in fact where its name comes from the Greek for sea monster. The hippocampus plays a role in memory that is as central as it is fascinating. The hippocampus is not, as we've established, the only place where memories are stored. But it is something like a coordinator. When a new experience enters the brain, the hippocampus receives signals from many different cortical regions, the sensory areas, the emotional areas, the areas that process time and place, and it begins the work of binding those signals together into a coherent, retrievable whole. Over time, through the process of consolidation, which accelerates during sleep, the hippocampus gradually transfers the memory to the cortex, where it can be stored more permanently. The hippocampus's direct involvement then becomes less necessary. The memory has been, in a sense, handed off. We know how critical the hippocampus is because of what happens when it is damaged. The most famous case in the history of neuroscience involved a man known for decades only as HM, whose hippocampus was surgically removed in an attempt to treat severe epilepsy. He could still remember everything that had happened before the surgery. But from that point forward, he was unable to form any new long-term declarative memories. Every new person he met, every new experience he had, faded within minutes as though it had never happened at all. He lived, for decades, in a permanent present, unable to move forward into memory, trapped forever at the edge of a moment that was always just slipping away. His life was one of the most important contributions to our understanding of what the hippocampus does. And in a quiet way, it is a reminder of how much of what we are is held together by structures we never think about, working gently, constantly, on our behalf, solidifying the days of our lives into something we can keep. And with that, we've arrived at the end of our first 10 facts. 10 quiet windows into one of the most astonishing things in the known universe, the way your brain holds on to the world. As you let these ideas rest, you don't need to remember them consciously. Your brain is already doing that work on your behalf. The hippocampus is already beginning its quiet labor. The circuits are already settling. All you need to do now is breathe. And let yourself drift a little further into the night. Welcome back to the quiet. If you're just settling in, finding a comfortable position, letting the weight of the day begin to release from your shoulders, from your jaw, from the small places where you've been holding tension without even noticing, that's exactly right. There's nothing to do right now but listen. The night is patient. And so are we. In our last segment, we began to sketch the broad landscape of memory, the sheer scale of what the brain can hold, the way memories are distributed rather than centralized, the three-stage journey every memory takes from experience to storage, and the remarkable role of the hippocampus in binding and solidifying what we live through. Tonight, we're going to go a little deeper. We're going to look at how the brain actually writes a memory. Not in abstract terms, but in something closer to the physical truth of it, the machinery, the chemistry, the almost impossibly small events that add up to the fact that you can close your eyes tonight and remember a face, a place, a feeling from 20 years ago. It's a gentle kind of wonder we're after. Not the sharp, electric kind. Just the low, warm kind. The kind that makes the world feel a little larger and a little softer at the same time. Let's begin where we left off. The hippocampus functions like a notebook, a temporary holding space, and over time, the notes it contains are transferred elsewhere and the pages, in a sense, are cleared. We spoke in our last segment about the hippocampus and its role as a coordinator, a binder of new experiences. And I used the image, briefly, of it handing memories off to the cortex over time. Let's stay with that idea a little longer, because there's something in it that is both scientifically rich and quietly beautiful. Think of the hippocampus as a notebook that you carry with you through your days. When something happens, When you meet someone new, when you learn something surprising, when an afternoon catches you off guard with its particular quality of light, the hippocampus writes it down. Quickly. In a kind of shorthand. It captures the outline, the key features, the emotional signature of the experience, and holds it ready for later processing. But the hippocampus isn't meant to be a permanent archive. It has a different job than long-term storage. And so, gradually, over days, weeks, months, and most efficiently during the deep hours of sleep, it transfers what it has written to the cortex. The memory becomes encoded more diffusely, more stably, across the wider brain. And the hippocampus, having passed the message along, is freed up. Its pages, in a sense, are cleared. Ready for tomorrow. This is why sleep matters so much to memory. This is why pulling an all-night and trying to retain what you studied rarely works as well as sleeping on it. The transfer, the consolidation, the clearing and rereading of the notebook, all of that happens most deeply in the dark, while you are still, while your conscious mind has stepped back from the world. Your hippocampus does some of its best work precisely when you think you're doing nothing at all. Memory storage is hierarchical, organized by layers of the brain, from older, deeper structures to newer, more recently evolved regions. The human brain is not a single, unified thing. It is, in a very real sense, several brains layered on top of one another, the product of hundreds of millions of years of evolution, each new capacity built on top of the old ones, the way a city builds new neighborhoods around an ancient center. The deepest structures, the brainstem, the cerebellum, are the oldest. They manage the fundamental, automatic rhythms of life, breathing, balance, the reflexes that keep you upright and alive. Above them sit the limbic structures, the hippocampus, the amygdala, which evolved later and handle emotion, motivation, and the kind of memory tied to experience and survival. And over all of this arches the neocortex, the wrinkled outer layer of the brain that is most elaborately developed in humans, which manages language, reasoning, planning, and the long-term storage of complex knowledge. Memory, it turns out, is stored hierarchically across these layers. Different levels of the brain hold different kinds of memory, and they do so with different degrees of permanence, different timescales of consolidation, different vulnerabilities to disruption. The layered architecture of the brain is not just an anatomical curiosity. It is the very structure within which memory lives. Understanding this hierarchy helps explain why some memories seem etched in stone while others dissolve overnight, why emotional memories can feel more vivid than factual ones, and why certain brain injuries erase some memories while leaving others completely untouched. Different layers of the brain hold different flavors of memory, from reflexive and emotional to factual and autobiographical. Following naturally from the hierarchy we just described, each layer of the brain specializes in a different flavor of what we call memory, and those flavors feel very different from the inside. The deepest layers handle the most automatic, the most ancient kinds of memory. The flinch away from something hot. The startle at a sudden sound. These are not memories you consciously recall, they are memories your body simply enacts, instantly, without consulting your higher reasoning at all. The middle limbic layers hold something warmer and more complex, emotional memory. The kind of memory that makes a particular song ache with association, or makes the smell of something cook on a stove reach all the way back to a kitchen from 30 years ago. These memories are not primarily verbal or conceptual. They are somatic, felt, visceral. They arrive as feeling before they arrive, if they ever do, as words. And the outer cortical layers hold the memories we think of as most purely cognitive, the facts, the narratives, the autobiographical stories we tell about ourselves. The carefully maintained sense of who we are and where we have been. All three flavors of memory are happening in you simultaneously, all the time. Right now, as you lie still, your brain is quietly tending to each of them, the automatic, the emotional, and the conscious, each in its own layer, each in its own time. And now we turn to the mechanics. To the question of how, physically, the brain actually writes a memory at all. Synapses are the physical sites where memories are stored. At the most fundamental level, the brain is a communication network. About 86 billion neurons, each one reaching out toward others with long, branching extensions, sending signals outward through structures called axons, and receiving incoming signals through a dense forest of smaller branches called dendrites. The point where one neuron communicates with another is called a synapse. And a synapse is not a point of contact, exactly. It is a gap, a tiny space, called the synaptic cleft, across which chemical messages are launched and received. One neuron releases molecules called neurotransmitters into that gap. The neighboring neuron catches them with specialized receptors. And the message, the signal, passes across. Now, here is the key. The strength of a synapse, how readily, how powerfully it transmits a signal, can change. Synapses are not fixed. They are plastic, in the technical sense of that word, capable of being shaped, strengthened, weakened, grown, or pruned in response to experience. And it is precisely in these changes, in the strengthening of certain synapses and the quieting of others, that memories are physically written. A memory is, at the molecular level, a pattern of synaptic changes. A landscape reshaped by experience. Every time something happens to you, the synapses that were active in processing that experience are nudged, however slightly, toward a new configuration. And in that new configuration, the ghost of the experience lives on. A single neuron can form thousands of synaptic connections, giving the brain an almost incomprehensible capacity for storing complex patterns. When you try to picture a neuron, you might imagine something fairly simple. A cell with a long tail, the axon, and perhaps a few branching arms. But the reality is far more elaborate than that. A typical neuron in the human brain may have anywhere from 1,000 to 10,000 synaptic connections. Some neurons, particularly certain types in the cerebellum, have been estimated to form connections with as many as 200,000 other cells. The dendritic branches are dense and elaborately structured, a miniature tree, with thousands of tiny protrusions called dendritic spines, each one hosting its own synaptic junction, its own little site where change is possible. When you multiply this across the 86 billion neurons in a human brain, the number of possible connections, the potential combinations, the potential patterns, reaches a number that has no meaningful comparison in the physical world. It is more than the number of stars in the observable universe. It is more than most of us can intuitively grasp. And every memory you have ever formed has altered some small subset of this vast network. Every experience has left its fingerprint somewhere in that incomprehensible web. The brain has room not only for everything you have ever lived, but for everything you ever could live. And still, in all likelihood, room beyond that. The cellular mechanism underlying memory formation is called long-term potentiation, or LTP. Now we arrive at one of the most important discoveries in the entire history of neuroscience. Long-term potentiation, LTP, is the process by which a synapse becomes stronger when it is repeatedly or intensely activated. And it is, as far as we currently understand, the fundamental cellular basis of learning and memory. The basic principle was articulated formally in the 1940s by the Canadian psychologist Donald Hebb, in a phrase that has since become one of the most repeated in all of neuroscience: neurons that fire together wire together. When two neurons repeatedly activate at the same time, the synapse between them grows stronger. The signal passes more easily, more reliably. The connection, in a very physical sense, becomes more deeply established. The discovery of LTP as an actual biological phenomenon, demonstrated experimentally in the 1970s, transformed the science of memory. Suddenly there was a mechanism, a measurable, reproducible, observable process that explained how experience could physically change the brain. How repetition could strengthen a skill. How emotion could carve a memory deeper than ordinary events. How the brain could learn. Long-term potentiation is not a metaphor. It is chemistry, physics, biology. It is the actual writing of experience into matter. NMDA receptors act as coincidence detectors. They only activate when two signals arrive at the synapse at precisely the same time. The mechanism of LTP depends on a specific type of receptor embedded in the membranes of neurons. It's called the NMDA receptor, named for a chemical used to study it, and it has a property that is almost philosophically elegant in its precision. The NMDA receptor will only allow signals to pass, under very specific conditions. It requires two things to happen simultaneously. The neuron sending the signal must be active, and the neuron receiving the signal must already be active, or recently active, at the same time. Both sides of the synapse must be engaged at once. This makes the NMDA receptor something scientists call a coincidence detector. It is a molecular gatekeeper that only opens the door when both conditions are met at exactly the right moment. If only one side is active, the receptor stays closed. Nothing changes. But when both fire together, in that precise alignment, the receptor opens, signals flow through, and the process of synaptic strengthening can begin. This mechanism is what gives LTP, and by extension, memory, its selectivity. Not just any activation strengthens a synapse. It has to be the right activation, in the right relationship, at the right time. The brain is not indiscriminately changed by everything that happens to it. It is changed by patterns, by co-occurrences, by the particular choreography of simultaneous experience. And the NMDA receptor is the molecular machinery that makes that discrimination possible. When NMDA receptors open, calcium flows into the cell, and it is this calcium influx that triggers the cascade of changes that eventually becomes a memory. When an NMDA receptor opens, one of the primary things that flows into the receiving neuron is calcium. Calcium ions, flooding inward through the newly opened receptor channel, and it is this flood, this specific chemical event at this specific location, that serves as the signal for the cell to begin changing itself. Calcium, inside the neuron, acts as a messenger. It doesn't just sit there, it sets off a cascade. It activates enzymes. Those enzymes trigger other enzymes. The cascade reaches into the cell's own internal machinery and begins to alter the structure of the synapse, making existing receptors more sensitive, moving additional receptors to the surface, and eventually, if the stimulation is strong or repeated enough, changing the actual physical shape of the dendritic spine that houses the synapse. The synapse, after a strong LTP event, is not the same synapse it was before. It has been physically remodeled. Its spine may have grown larger. Its receptor complement may have changed. It transmits signals more readily, more reliably, than it did before the experience that triggered all of this happened. All of that, from experience to changed synapse, begins with calcium. A tiny ion. An almost impossibly small messenger. Carrying, in its momentary flood, the instruction to remember. Memory also depends on molecular switches at synapses, proteins that can exist in an on or off state and help maintain a memory over very long timescales. We have a problem, though. And it is a problem that took scientists a long time to understand how biology solves. Proteins don't last forever. The proteins that make up a synapse are constantly being broken down and replaced, on timescales of hours to days. And yet memories can last for decades. How can a memory survive when the very molecules that encode it are being continually turned over? Part of the answer lies in what are called molecular switches, proteins that exist in two stable states, on and off. When a synapse is strengthened during LTP, certain of these switch proteins are flipped from their off state to their on state. And crucially, once flipped, they tend to stay on. They can even flip neighboring switch proteins to the on state, propagating and maintaining the active configuration over time. One of the most studied of these switches is a protein called pKm-zeta, though the full story of exactly how it works is still being refined by ongoing research. What matters for our purposes is the broader principle. The brain has evolved molecular mechanisms specifically designed to maintain synaptic changes over very long timescales, despite the constant turnover of the proteins involved. The switch can be reset again and again with fresh proteins, but the on state persists. The memory survives its own molecular substrate. The river keeps flowing even as the water in it is always changing. New protein synthesis is required for long-term memory. Without the creation of new proteins, memories cannot be permanently stored. If you want a memory to last, not just for minutes or hours, but for days, years, decades, the brain must do something more than simply alter the state of existing proteins at a synapse. It must build new ones. This is one of the more striking findings in the cellular biology of memory. Long-term memory formation requires protein synthesis. New proteins must be manufactured. The cell's genetic machinery, the nucleus of the neuron, deep inside, must be activated to produce the molecular building blocks needed to permanently restructure the synapse. Scientists have demonstrated this elegantly by using drugs that block protein synthesis in experimental animals. When these drugs are administered immediately after a learning experience, the animals can still form short-term memories. They can remember what just happened. But long-term memory fails to form. The experience doesn't consolidate. The memory doesn't stick. Hours later, it is as if the learning never happened. This has real implications for how we think about memory consolidation. It suggests that there is a window, a period of time after a new experience during which the memory is still being actively constructed at the molecular level. A window during which it can be supported or disrupted. And it is another reason why sleep matters so much. Many of the processes of protein synthesis that support memory consolidation run most efficiently during sleep, when the brain's resources are not being directed outward toward the world, but inward toward the quiet, essential work of building permanence out of experience. And so we've arrived, quietly, at the end of our second segment. We've traveled from the broad architecture of memory down into the microscopic, into the gap between two cells, into the chemistry of a single receptor, into the flood of a tiny ion that sets in motion the writing of something you will carry with you always. It is, if you let yourself feel it, a remarkable thing. Every experience that has ever mattered to you has left a physical trace, has moved calcium across a membrane, has activated enzymes, has grown new proteins, has, in the most literal sense, changed the shape of the matter inside your skull. You are not unchanged by what you have lived. You are written by it. Every memory is, in the deepest possible sense, a part of the structure of who you are. Let that sit with you now. There's nothing more you need to do. Just let it settle, the way things settle in the dark, slowly, gently, into place. Rest now, and we'll continue when you're ready. Welcome back to the stillness. If you're returning after some sleep, or if you've been drifting in and out, that's perfectly fine. That's actually, as you'll come to appreciate even more by the end of tonight's segment, exactly what your brain needs. The quiet intervals are not empty. They are full of work you cannot feel, the gentle, persistent labor of consolidation, of proteins being built, of synapses being settled into their new configurations. We've come quite a long way already. We started with the grand scale, the almost incomprehensible storage capacity of the brain, the distributed nature of memory, the three-stage journey from encoding to retrieval. Then we went smaller, down into the cellular mechanics, synapses and LTP and NMDA receptors and the flood of calcium that begins the writing of a lasting memory. Tonight, we go smaller still. For a while. We're going to spend some time at the molecular level, the level of proteins and genetic machinery and the structure of DNA itself. And then, at the end, we're going to pull back and return to the wide view, to the cinematic scale, and look at how memory spreads itself across the whole landscape of the brain. It is a journey inward and then outward again. And it's one worth taking slowly. So settle in. Let your breathing find its own rhythm. And let's begin. Molecular tags are used by neurons to direct newly synthesized proteins to exactly the right synapses, ensuring that the changes made during learning are targeted precisely. We established in our last segment that long-term memory requires the synthesis of new proteins. The genetic machinery of the neuron must be switched on, new proteins must be manufactured, and those proteins must be incorporated into the synapses that were active during the original experience. But here is where things get beautifully intricate. A neuron might have thousands of synapses. Tens of thousands, in some cases. When new proteins are manufactured in the cell body, deep at the center of the neuron, how do they know which of those thousands of synapses to travel to? How does the right protein end up at the right location? The answer lies in what scientists call synaptic tagging. When a synapse undergoes strong activation, the kind of activation associated with learning, it generates a temporary molecular tag. A flag, in effect, raised at that specific location. When new proteins are subsequently synthesized and travel outward through the neuron's branching structure, the tagged synapses capture them. The flag says, "Here. Come here." This is the one that needs reinforcing. The tag itself is temporary. It lasts for perhaps an hour or two, which creates a window during which events at other synapses can interact with the process, a phenomenon with its own fascinating implications for how memories are colored by context and association. But the core principle is elegant. The brain has evolved a precise molecular addressing system so that the changes made during learning are not scattered randomly, but are delivered with remarkable specificity to exactly the right physical location. Your brain, even at the molecular level, knows where to send the message. 2. Memory storage is supported by physical, structural changes to dendritic spines, the tiny protrusions that host synaptic connections. We've talked about chemistry, about signals and cascades and protein synthesis. But memory also changes the brain in a way that is, in a sense, more tangible than any of that. It changes the physical shape of neurons. The synapses on a neuron are hosted on tiny protrusions that extend from the dendrites, the receiving branches of the cell. These protrusions are called dendritic spines. And they are not static, fixed structures. They are dynamic, changing things, capable of growing larger or smaller, appearing or disappearing, being remodeled in response to experience. When a synapse is strengthened through LTP, its dendritic spine often grows. It becomes larger, more robust, with a bigger head and a thicker neck. The physical increase in size corresponds to an increase in the number of receptors it can accommodate, and therefore in the strength of the signal it can receive and transmit. A large, mature spine is a synapse that has been exercised, that has been reinforced by experience. This physical remodeling is one of the reasons why memories are so durable. It is not only chemistry, it is architecture. The brain literally reshapes itself to hold on to what it has learned. New experiences build new structures. Repeated experiences enlarge and stabilize existing ones. And over time, The physical landscape of your neural tissue comes to reflect the accumulated weight of everything you have experienced and learned and remembered. You are, in the most literal possible sense, architecturally shaped by your life. Memory involves multiple phases of synaptic plasticity: an early, fast phase that requires no new proteins, and a later, slower phase that requires full-scale synthesis. Not all memory formation happens at the same speed, or by the same mechanism. Scientists have identified distinct phases of synaptic plasticity, distinct stages in the process by which a synapse is changed by experience, and they operate on very different timescales and by very different cellular means. The first phase is early. It happens quickly, within minutes of a strong experience. In this early phase, existing proteins at the synapse are modified, their shape or position or activity is changed, but no new proteins need to be built. It's a rapid response, like rearranging furniture that's already in the room. This early phase can produce a form of short-term synaptic strengthening that might last minutes to hours. The second phase is late, and it's the one that matters for lasting memory. This phase requires the activation of gene expression. The cell's nucleus must be engaged, new proteins must be manufactured and shipped out to the synapse. This phase is slower, taking hours to fully develop. But it produces changes that are far more durable. The synapse is not just rearranged, it is rebuilt. New materials are added. New structures are formed. The distinction between these two phases has real consequences. It is why you can remember something vividly in the moment, but wake up the next morning to find it has faded. The early phase fired, but the late phase, for whatever reason, perhaps fatigue, perhaps insufficient emotional salience, perhaps the disruption of sleep, did not consolidate fully. The furniture was moved, but the new room was never quite built. Memories are stabilized by feedback loops that run all the way back to the nucleus of the neuron, ensuring the cell knows which genes to keep active. Here is something that might sound almost too elegant to be true, but is borne out by decades of research. The process of memory consolidation involves feedback loops. Signals generated at the synapse, out at the very tips of the neuron's branches, perhaps millimeters away from the cell body, travel all the way back to the nucleus, where they influence which genes are expressed and for how long. This means that what happens at the periphery of the cell, the strengthening of a synapse by experience, reaches back to the very center of the cell's genetic operations and leaves an instruction there. Keep these genes active. Continue producing these proteins. Maintain this configuration. It is a communication system of extraordinary subtlety. The synapse tells the nucleus what kind of experience just happened. The nucleus responds by producing the molecular resources needed to make that experience last. The new proteins travel back out to the synapse. And the synapse, reinforced, sends further signals back to the nucleus to maintain the process. Back and forth. Synapse to nucleus, nucleus to synapse. A conversation between the periphery and the center, maintaining the structural change, keeping the memory alive. The brain is not passive in the face of experience. It responds to it, actively, at every level of its organization, from the molecular to the architectural, from the moment of experience to the long, quiet years that follow. Certain proteins act as dedicated memory switches in specific neural circuits, meaning different circuits use different molecular machinery to remember. We have spoken of the brain as many memory systems running simultaneously, different circuits for different types of memory, each one specialized in the kind of recollection it handles. And this specialization goes all the way down to the molecular level. Different neural circuits use different proteins as their primary memory switches. The molecular machinery that stabilizes a memory in the hippocampus is not identical to the machinery that stabilizes a motor memory in the cerebellum, or an emotional memory in the amygdala. Each circuit has its own molecular vocabulary, its own preferred proteins, its own particular cascade of events that consolidates experience into something lasting. This has been one of the more humbling discoveries in the biology of memory, the realization that there is not one universal mechanism, a single master switch that the whole brain uses. Instead, there is a family of related mechanisms, each one adapted to the specific demands of its circuit, each one shaped by the evolutionary pressures that built that particular part of the brain for that particular purpose. It means that understanding memory fully requires understanding each of these systems separately, and then understanding how they interact. It is, as one might expect of something as rich and varied as the experience of remembering, more complex than any single story can capture. But the complexity is the point. The complexity is what allows a single brain to hold everything from muscle memory to mathematical knowledge to the particular sadness of a Tuesday afternoon from 30 years ago. The molecular machinery of memory reuses common signaling toolkits. The same basic components appear again and again across different memory systems. And yet, for all the variation we just described, there is also a deep conservation. The different memory systems of the brain, despite their differences, share a common molecular heritage. They draw from the same toolkit. The same families of enzymes, kinases, phosphatases, the proteins that modify other proteins, appear across many different memory circuits. The same second messenger molecules, calcium, cyclic AMP, others, serve as internal signals in many different contexts. The same downstream targets in the nucleus, the same transcription factors that switch genes on and off, appear again and again across species and circuits and types of memory. This conservation tells a story about evolution. The basic molecular machinery of synaptic plasticity is ancient, far older than the mammalian brain, older even than vertebrates. It was found to work by the long process of natural selection, and it was kept. Built upon, specialized, adapted, but kept. The worm and the fly and the mouse and the human all share versions of the same basic memory machinery. This is not just a curiosity. It is, in practical terms, part of why research on simpler organisms has illuminated so much about the human brain. When a scientist discovers how a particular enzyme shapes memory in a sea slug, and some of the most foundational discoveries in memory biology came from exactly that kind of work, they are often discovering something that applies, with modifications, all the way up the evolutionary tree. The toolkit is shared. And that shared toolkit is part of what makes memory, in its essentials, one of the oldest and most fundamental capacities of living things. The way DNA is packaged inside the neuron's nucleus can change in response to experience, and these packaging changes affect which memory-related genes can be accessed. Now we arrive at something that would have seemed almost impossible to scientists a generation ago. We are going to talk about how memory reaches all the way down to the DNA itself, not by changing the sequence of the genetic code, but by changing how that code is physically organized and accessed. DNA, inside every cell in your body, is not simply floating free. It is wrapped around proteins called histones, coiled and packaged into a dense structure. And how tightly or loosely the DNA is wound around these proteins determines whether the genes embedded in it are accessible, whether the cell's machinery can read them, activate them, and produce the proteins they encode. Experience, it turns out, can change this packaging. Neural activity associated with learning and memory can cause certain regions of the DNA to be unwound, opened up, made more accessible, while others are more tightly coiled and silenced. These changes in packaging alter which genes are available to be expressed, and for how long. This is a physical change in the structure of the material inside the nucleus of your neurons. Not a change in the genetic sequence, your DNA letters remain the same. But a change in the three-dimensional shape of the library, in which books are shelved in accessible places and which are kept behind locked doors. Experience rearranges the library. And in doing so, it shapes which molecular resources are available for the work of memory. Epigenetic marks, chemical tags added to DNA or to histone proteins, help prime memory-related genes, keeping them ready for activation when needed. The packaging changes we just described are part of a broader system that scientists call epigenetics, a word that means, roughly, above or around the genetics, referring to modifications that don't alter the sequence of DNA but profoundly affect how it is used. Specific chemical groups can be added to or removed from the histone proteins around which DNA is wound, and these additions and removals act as signals, tags that tell the cell's machinery whether to open or close a particular stretch of DNA, whether to activate or silence the genes within it. Some of these epigenetic marks are associated with memory. When an animal undergoes a significant learning experience, epigenetic marks appear at specific locations in the genome, marks that prime the memory-related genes, keeping them in a ready state, poised for activation the next time a relevant signal comes in. The gene hasn't been turned on yet. But it is, in a sense, at the ready. Leaning forward. Waiting for the word. These marks can persist for a remarkably long time, days, weeks, in some cases potentially much longer. They represent a form of cellular memory that outlasts any individual protein, maintaining a kind of molecular readiness in the neuron long after the original experience. The gene has not forgotten. The mark is still there. The three-dimensional structure of DNA inside the nucleus changes with learning. Certain memory-related genes are physically repositioned to become more accessible. We go even deeper now. Not just to the surface of the DNA, where epigenetic marks are added, but to the full three-dimensional architecture of how DNA is organized within the nucleus. The nucleus of a cell is not simply a bag full of tangled DNA. The genetic material is organized in space, Different chromosomes occupy different regions of the nucleus, and genes are spatially arranged in ways that affect their activity. Genes that are frequently expressed tend to be found in the more active, accessible regions of the nuclear interior. Genes that are silenced tend to be sequestered toward the periphery, against the inner wall of the nucleus. Recent research has shown that learning can cause certain genes to be physically relocated within the nucleus, moved from the periphery toward the more active interior, repositioned closer to the molecular machinery that reads and transcribes them. The three-dimensional geography of the genome changes in response to experience. This is perhaps the deepest level at which memory has been found to operate, not just at the synapse, not just in the cascades of proteins and enzymes, not just in the epigenetic marks on the surface of DNA, but in the actual spatial arrangement of genetic material inside the cell. Memory changes the shape of the library, the positions of the books within it, and the accessibility of the shelves. It reaches, in the most literal sense imaginable, all the way to the core. And now, having gone as deep as we can go, having descended through the synapse, through the protein cascades, through the epigenetic marks, all the way to the three-dimensional architecture of the genome inside a single cell, it is time to rise back up. To pull back. To widen the lens. To look at the whole brain again. Engram cells, the cells that store a specific memory, are not clustered together in one spot. They are distributed across multiple brain regions simultaneously. We have come full circle to the engram. To those tagged cells, those memory-bearing neurons, that we first encountered in our opening segment. And now, having understood something of the molecular machinery that makes a cell part of a memory, we can ask a question we weren't quite ready for then. Where, exactly, are the engram cells for any given memory? The answer is remarkable. They are everywhere. Or rather, they are distributed. Spread across multiple brain regions, each cluster contributing a different dimension of the memory, none of them alone sufficient to hold the whole thing. For a rich autobiographical memory, a specific event, with its visual details and its sounds and its emotional texture and its spatial and temporal context, the engram cells are scattered. Some are in the visual cortex. Some are in the auditory cortex. Some are in the prefrontal cortex, holding the narrative structure of the event. Some are in the amygdala, holding its emotional weight. Some are in the hippocampus, holding the binding information that allows all the other cells to be coordinated and recalled together. The memory does not live in any one of these populations of cells. It lives in the relationship between them, in the pattern of connections that links them, in the learned tendency of these distributed cells to fire together when the memory is triggered. Like a constellation. Like a chord. No single star is the constellation. No single note is the chord. The memory is the pattern. The memory is the whole. And the whole is spread, quietly, across the vast and extraordinary landscape of the brain that is right now, as you lie still and listen, holding everything that you are. We've covered a great deal of ground tonight, from the molecular addressing systems that direct proteins to the right synapses, through the physical remodeling of dendritic spines, through epigenetic marks and the three-dimensional architecture of DNA, and finally back out to the wide view, to engram cells spread across the whole brain like stars in a private sky. It is a lot to hold. And you don't need to hold it consciously. That's not what this is for. These ideas are here to drift through you, the way water moves through stone over a long time, not with force, but with patience. Leaving something behind that you couldn't quite name, but that changes the shape of things nonetheless. Your brain, right now, is doing exactly that. Taking what it has encountered tonight and beginning, slowly, the long work of making it part of you. Let it work. You just rest. Welcome back to the quiet. However you've arrived here tonight, whether you've been listening from the very beginning, or you've drifted off and returned, or this is the first segment you're finding your way into, you are welcome here. There is no wrong way to do this. The voice will continue at the same unhurried pace regardless. The night is long and generous, and there is nowhere else to be. In the segments we've shared so far, we've moved through a remarkable amount of territory. We began at the very largest scale, the near-infinite capacity of the brain, the distributed nature of memory, the broad systems and regions that different types of memory call home. Then we descended into the cellular world, into synapses and receptors and the cascade of molecular events that physically writes an experience into the structure of a neuron. And in our last segment, we went even deeper into protein tagging, epigenetic marks, and the three-dimensional architecture of DNA itself, before pulling back to the wide view of engram cells scattered like constellations across the whole brain. Tonight, we're going to stay at that intermediate scale. Not quite the molecule, not quite the whole brain, but the level of circuits, of networks, of the rhythms and oscillations that run through the brain like tides. We're going to look at memory not as a fixed thing that gets stored and then simply sits there, but as something alive, dynamic, continuously being woven and rewoven across time. Because it turns out that memory is not a destination. It is a process. And that process never really stops. Memory is physically rewired across the days and weeks following an initial experience. The brain continues actively restructuring itself long after the moment of learning. There is a tendency to think of memory formation as something that happens quickly, at the moment of experience, or perhaps over a single night of sleep. And there is truth in that picture, as far as it goes. But it is only part of the story. The full process of memory consolidation, the real, deep embedding of an experience into the brain's long-term architecture, unfolds across a much longer timescale than a single night. In the days and weeks following a significant experience, the brain continues to rewire itself. Synaptic connections are further strengthened or pruned. The balance of which regions are involved in storing and retrieving the memory gradually shifts. The hippocampus, which played such a central role early on, begins to hand its responsibilities over to the cortex. And the cortical representation of the memory, the distributed network of engram cells spread across sensory and association regions, becomes progressively more stable, more self-sufficient, less dependent on hippocampal coordination. This extended rewiring is sometimes called systems consolidation, to distinguish it from the faster synaptic consolidation that happens at the cellular level. And it means that a memory is not fully formed on the day you acquire it. It is still being shaped days later. still being integrated and restructured weeks later. The physical changes in the brain that represent a new memory continue to unfold long after the experience itself has ended. In a very real sense, the brain is always a few weeks behind, always still digesting the recent past, always still deciding, in its slow and patient way, what is worth keeping and how it should be kept. Memory consolidation is highly dynamic. Memories can be updated, integrated with new information, and even partially rewritten each time they are recalled. This is one of the most quietly unsettling, and also one of the most fascinating, things we know about memory. Every time you recall a memory, you are not simply playing back a fixed recording. You are reconstructing it, reassembling it from its distributed pieces. And in the act of reassembly, the memory becomes temporarily vulnerable, temporarily plastic again, open to change. This phenomenon is called reconsolidation. When a memory is retrieved, the synaptic connections that hold it enter a briefly destabilized state, a window during which new information can be incorporated, errors can be introduced, emotional associations can be updated. The memory is then restabilized, reconsolidated, in its new form. This is not a flaw in the system. It appears to be a feature. Reconsolidation allows memories to be updated in light of new experience, allows the past, in a sense, to be gently revised to stay useful in the present. If you learn something new that modifies your understanding of an old event, reconsolidation allows the old memory to incorporate that new understanding, to become more accurate or more contextually appropriate. But it also means that memories are not perfectly reliable recordings. They are living documents, subject to revision. Every time you remember something, you are also, subtly, remembering it slightly differently than you did before. The memory of the memory becomes part of the memory itself. This is, if you let yourself sit with it, a rather profound thing. The past is not fixed. It is continuously, quietly, being rewritten by the person you are becoming. Thalamocortical circuits, the looping connections between the thalamus and the cortex, help fine-tune and stabilize cortical memory representations over time. Deep in the center of the brain, nestled between the cortex above and the brainstem below, sits the thalamus. It is sometimes described as a relay station, a central hub through which sensory information passes on its way to the cortex. And that is true, as far as it goes. But the thalamus is more than a relay. It is an active participant in the processing and shaping of the signals it handles. Between the thalamus and the cortex there are loops, circuits that run upward from thalamus to cortex and back down again, continuously cycling signals back and forth. These thalamocortical loops are involved in many functions, in the regulation of sleep and wakefulness, in the gating of sensory information, in the coordination of cortical activity across different regions. And they appear to play a role in the stabilization of cortical memory representations. As memories migrate from hippocampal dependence toward cortical independence, the long process we described in the previous fact, the thalamocortical circuits help tune that cortical representation. They help coordinate the activity across the distributed network of cortical engram cells, reinforcing the connections between them, gradually ensuring that the network can be reactivated reliably without needing the hippocampus to hold it all together. The thalamus, in this picture, is something like a conductor, ensuring that the various instruments of the cortical orchestra are playing in time with one another, that the memory, when recalled, sounds like a coherent whole rather than a collection of disconnected fragments. Memory integration happens across time. New experiences are not stored in isolation, but are woven into existing networks of related knowledge and past experience. No memory is an island. Every new experience you have is not stored in empty space. It is stored in a brain that already contains a vast and richly interconnected network of previous experience. And the new memory is integrated into that network, linked to what came before, connected by association and similarity and emotional resonance to memories that are sometimes decades old. This integration is part of what makes memory so useful. A memory that is connected to many other memories, that has been woven deeply into a rich associative network, is more easily retrieved, more flexibly applied to new situations, and more readily combined with other knowledge to produce insight. The more a new memory connects to what you already know, the more likely it is to stick, and the more likely it is to be genuinely useful rather than merely stored. This is part of why learning something new in a context rich with prior knowledge is easier than learning the same thing in isolation. The new knowledge has more hooks to catch on, more existing connections to anchor itself to. The network is receptive. The integration is easier. And it means that the brain's memory networks are not simply growing larger over time, like a library adding new books to empty shelves. They are growing more richly interconnected, more deeply cross-referenced, more elaborately associated. Every new memory subtly reorganizes the network it enters. It doesn't just take up residence. It changes the neighborhood. Different types of memories use distinct circuit-specific stabilization pathways. The route by which a memory becomes permanent depends on what kind of memory it is. We have returned, again, to the theme of diversity within the memory system. And it applies here too, to the process of stabilization itself. The pathway by which a declarative memory, a conscious, reportable memory of an event or a fact, becomes stable over time is not the same pathway used by a procedural memory, a habit, a motor skill. The hippocampal cortical transfer that we've spent so much time describing is primarily relevant for declarative memory. Procedural memories, consolidated through the basal ganglia and the cerebellum, follow different stabilization routes, on different timescales, with different vulnerabilities and different requirements. Emotional memories, consolidated with heavy involvement from the amygdala, follow yet another pathway, one that tends to produce memories that are extraordinarily vivid and durable, because the amygdala's involvement during emotional arousal triggers additional consolidation signals that essentially tell the rest of the brain, "This was important, make sure it sticks." This is one of the reasons why emotionally charged events are so much better remembered than emotionally neutral ones. It is not simply that you were paying more attention. It is that the amygdala was actively amplifying the consolidation signal, ensuring that the memory received extra resources during the stabilization process. Your brain has a built-in mechanism for prioritizing what matters. And matters, to your ancient biology, is closely correlated with what feels. Memory storage is partly shaped by network oscillations, rhythmic waves of electrical activity that sweep through the brain and help coordinate the timing of consolidation. the brain is never electrically still. Even in the deepest sleep, even in the most restful moments of quiet wakefulness, electrical activity sweeps through the neural networks in rhythmic waves. These oscillations, measurable with an EEG, visible as characteristic waveforms on a display, are not simply background noise. They are part of the brain's operating system. They coordinate activity across distant regions. They gate the flow of information. And they play a direct role in memory consolidation. Different oscillation frequencies are associated with different brain states and different cognitive functions. Gamma oscillations, fast and high frequency, are associated with active processing and the binding of information across regions. Theta oscillations, slower and rhythmic, are strongly associated with hippocampal activity during learning and navigation. And the slow oscillations of deep sleep, the great rolling waves that sweep across the cortex during non-REM sleep, appear to be particularly important for the transfer of memories from hippocampus to cortex. The timing matters. Memory consolidation is not just about what happens at the synapse, it is about when it happens, in relation to the rhythmic tide of the brain's electrical activity. Synaptic changes that are timed to the peaks of particular oscillatory cycles are more likely to be stabilized than those that occur out of phase. The oscillations, in a sense, provide the rhythm to which the molecular machinery of memory must learn to dance. Memory consolidation is supported by distributed replay, the reactivation of memory-related neural patterns across multiple brain regions simultaneously, not just in the hippocampus. During sleep, the brain replays the events of the day. We have touched on this before, but let's linger here a little longer, because the nature of that replay is more remarkable than it might first appear. For a long time, replay was understood primarily as a hippocampal phenomenon, the hippocampus reactivating the patterns it had recorded during waking experience, running through them again, reinforcing them. And hippocampal replay is real and well documented. But research over recent years has revealed that the replay is far more distributed than that. When the hippocampus replays a memory pattern, it simultaneously reactivates corresponding patterns in the cortical regions that were originally involved in that experience. The visual cortex replays the visual components. The auditory cortex replays the sounds. The prefrontal cortex replays the narrative structure. The whole distributed network of engram cells fires together again, not as powerfully or as richly as during the original experience, but in the same coordinated pattern. This distributed replay serves to strengthen the connections between the engram cells spread across these different regions. Each replay episode is another rehearsal of the full pattern, another reinforcement of the connections between the nodes of the network. Over hundreds of replay episodes across many nights of sleep, these connections become progressively more robust, more efficient, more capable of being triggered by a partial cue and reconstructing the whole. Sleep, in this picture, is not merely rest. It is rehearsal. A quiet, invisible, involuntary rehearsal of everything that happened today, running through the night, building the architecture of tomorrow's memory. Some of the most important memory replay during sleep happens silently, without detectable neural firing, through subtle changes in synaptic weights that occur below the threshold of measurable activity. Here is something that might feel, at first, almost paradoxical. We just described memory replay as a process of reactivation of neural patterns firing again during sleep. But some scientists have proposed, and accumulated evidence suggests, that an important component of consolidation during sleep may happen without overt neural firing at all. This idea is associated with what is sometimes called synaptic homeostasis, the proposal that during sleep, the brain doesn't only replay and strengthen memory traces, but also globally downscales synaptic strengths, bringing the overall level of synaptic excitability in the brain back to a sustainable baseline. During waking, many synapses are strengthened by experience. During sleep, some of that strengthening is gently dialed back, not erased, but normalized, so that the brain doesn't become progressively more overexcited over time. Within this process of downscaling, the memories that were most strongly encoded during waking, the ones with the most robust initial traces, survive the downscaling better than weaker traces. They are, relatively speaking, left stronger. The signal, you might say, is preserved while some of the noise is quietly reduced. And some of this normalization may happen not through replay-driven firing but through slow, subtle shifts in the molecular state of synapses, changes that occur below the threshold of detectable electrical activity, invisible to standard recording methods, but real in their molecular consequences. The brain consolidates memory, in other words, not only by actively replaying the past, but also by quietly, imperceptibly, tidying the landscape in which the past is stored. Sleep actively strengthens memory traces. People who sleep after learning remember significantly more than those who remain awake for the same period. This is one of the most consistently replicated findings in all of memory science, and it is worth stating simply and clearly: sleep helps you remember. Across hundreds of studies, in many laboratories, using many different kinds of material, words, faces, spatial layouts, motor skills, emotional associations, the same finding emerges. People who sleep after a period of learning retain more than people who remain awake for the same interval. The gap is not small. In many studies, it is dramatic. Sleeping is not merely better than staying awake. It is actively beneficial in a way that no amount of quiet wakefulness can replicate. This is not simply because sleep prevents interference, though that is part of it, since during sleep you are not encountering new experiences that might compete with or overwrite what you learned. It is because sleep actively drives the consolidation processes we have been describing, the replay, the synaptic remodeling, the protein synthesis, the transfer from hippocampus to cortex. Sleep doesn't just passively preserve the memory. It builds it. And different stages of sleep appear to support different types of memory. REM sleep, the stage associated with vivid dreaming, appears particularly important for emotional memories and for the flexible integration of new information with existing knowledge. Non-REM sleep, and particularly the deep slow-wave sleep that dominates the early part of the night, appears most important for the consolidation of declarative memories and for the hippocampal cortical transfer. A full night of sleep, with its natural cycling through these stages, is not a luxury. It is, from the perspective of your memory system, an essential part of the learning process itself. Sleep doesn't just strengthen memories as they are, it also reshapes and generalizes them, extracting patterns and insights that weren't consciously accessible during waking. And here is perhaps the most remarkable thing of all about sleep and memory. Sleep does not simply preserve what you learned. It transforms it. There's a phenomenon that researchers have documented in multiple contexts. People who sleep after exposure to a complex problem or a body of information sometimes wake up having gained insight that they did not have when they fell asleep. The brain, during sleep, appears to do something more than simple consolidation. It appears to work on the material, to abstract from it, to find patterns, to extract the underlying structure and make it more generally applicable. In one classic experiment, participants were taught a mathematical task that could be solved step by step, but which had a hidden shortcut, a pattern that, if noticed, would make the whole thing much faster. Most participants who stayed awake did not discover the shortcut on their own. But those who slept after learning were dramatically more likely to wake up and spontaneously apply it. Sleep had, in some way, reorganized their knowledge of the task and made the hidden structure visible. This is what it means to sleep on a problem. The phrase, "it turns out," is not merely a figure of speech. It is a description of something real that the brain does, something that waking cognition, for all its remarkable capabilities, cannot fully replicate. Sleep takes the raw material of experience, the particular and the specific, and quietly draws from it something more general. Something that can be applied not just to what happened today, but to whatever tomorrow brings. It is the brain at its most creative, not in the flashy, deliberate way we usually associate with creativity, but in the deep, slow, patient way of something that has been doing this for a very long time and has gotten rather good at it. We have traveled, in this segment, from the extended timescale of memory rewiring, the weeks-long process of consolidation that continues long after an experience ends, through the dynamic, revisable nature of memory, through thalamocortical circuits and network oscillations and distributed replay, through the silent, below-threshold processes of synaptic normalization, and finally to the transformative power of sleep. And what emerges from all of this, if you hold it gently and don't try to grip it too tightly, is a picture of memory not as a static archive but as a living, breathing, continuously active process. The brain is always working on the past. Always revising, integrating, stabilizing, generalizing. Always finding, in the material of what has been, the seeds of what might be understood more deeply tomorrow. And it does much of its best work exactly now. In the dark. In the stillness. While you let go of the day and allow the night to do what nights are for. Your brain knows what it's doing. You can trust it. You can let everything that needs to be held, without any effort on your part at all. Just breathe. And rest. Welcome back to the stillness. If you've been here since the beginning, you've now traveled quite a distance through the landscape of memory. From the vast scale of the brain's storage capacity down to the molecular machinery of a single synapse. From the three-stage life cycle of encoding, consolidation and retrieval, all the way through the rhythmic oscillations of sleeping brain waves and the quiet, invisible work of nightly replay. It has been, in the best possible sense, a lot. And none of it requires your conscious effort to hold. It is already, in whatever form it will take, beginning its slow journey into the architecture of you. Tonight we arrive somewhere different. Somewhere that might feel a little closer to home. Because the facts in this segment are not primarily about molecules or circuits or the technical vocabulary of neuroscience. They are about something more immediately recognizable. They are about why certain things stick and others vanish. Why you can remember, in vivid detail, the moment you heard a piece of news that changed everything, but cannot remember what you had for lunch 3 Tuesdays ago. Why fear carves memory deep. Why surprise opens a door that ordinary experience cannot. This is the segment where the science becomes, unmistakably, about being human. So settle in. Let your body be heavy. Let the breath come and go without any management from you. And let's begin. Attention is the gatekeeper of memory. Without it, experience passes through the brain without leaving a lasting trace. There is an experiment that has become something of a classic demonstration in the psychology of attention. Participants are shown a video of people passing a basketball back and forth, and are asked to count the number of passes. Halfway through the video, a person in a gorilla suit walks into the frame, pauses in the center, thumps their chest, and walks off. And when participants are asked afterward whether they noticed anything unusual, a significant proportion of them, minds fully occupied with counting passes, saw no gorilla at all. The gorilla was there. It was large and obvious and walked through the middle of the scene. But because attention was elsewhere, it left no trace. This is attention acting as gatekeeper. Memory does not record everything that happens to you. It records what you were paying attention to. The rest, the vast, continuous flood of sensory information that washes over you every waking moment, is filtered, reduced, and largely discarded before it ever has the opportunity to become a lasting memory. Attention determines what gets through the gate. And what gets through the gate is, with relatively few exceptions, what you will later be able to remember. Everything else, however real and present it was in the moment, simply passes through without leaving a mark. This is not a failure of the memory system. It is, in fact, one of its most important features. A brain that tried to remember everything, every face glimpsed on a busy street, every word of background conversation, every flicker of light on every surface, would rapidly become unusable. Selectivity is not a limitation. It is the mechanism that makes memory coherent, navigable, and meaningful. But it does mean that what you remember about your life is, to a significant degree, a portrait of what you chose, or were caused, to attend to. The memories you carry are, in the deepest sense, a record of where your attention has lived. Memory encoding is heavily influenced by both attention and emotion. The combination of the two produces the most durable memories the brain is capable of forming. Attention is necessary for memory. But it is not sufficient. You can pay careful, deliberate attention to something and still forget it within hours if it carries no emotional weight. And you can be taken entirely by surprise by something, can have your attention grabbed rather than given, and find that the memory it leaves is among the most vivid you possess. The second great shaper of encoding, alongside attention, is emotion. And the two interact in ways that are deeply important to understand. Emotion amplifies encoding. When an experience carries emotional charge, when it frightens you or moves you or fills you with sudden joy or grief or awe, the brain responds differently than it does to neutral experience. The amygdala, that almond-shaped structure nestled deep in the temporal lobe and so closely associated with emotional processing, becomes highly active. And when the amygdala is active during an experience, it sends signals to surrounding memory structures that essentially say, pay extra attention to this. Use more resources. Make this one last. The result is what researchers call emotionally enhanced memory. Emotional experiences are encoded more deeply, more richly, and with more redundancy than emotionally neutral ones. They are more likely to survive the night. More likely to still be vivid a week later, a year later, a decade later. The combination of full attention and strong emotion is, neurologically speaking, the most powerful memory-forming state the brain can enter. When both are present simultaneously, when you are fully present to something that also matters deeply, the encoding that results is as close to indelible as human memory gets. Working memory, the system that holds information in mind for immediate use, has a remarkably small capacity, typically limited to around 4 items at a time. Here is something that might feel, at first, a little deflating. For all the vast and almost incomprehensible storage capacity of long-term memory that we described in our opening segment, the petabytes of potential information, the hundreds of years of potential footage, the workspace that your brain uses for immediate, conscious, moment-to-moment thinking is tiny. Working memory is the system that holds information actively in mind, the mental scratchpad you use when you're doing arithmetic in your head, or holding the beginning of a sentence in mind while you read to its end, or keeping track of several things at once during a complex task. And its capacity is severely limited. For most of the second half of the 20th century, the accepted figure was 7 items, plus or minus 2, a number proposed by the psychologist George Miller in the 1950s. More recent research has revised this estimate downward. The current consensus is closer to 4 chunks of information, and sometimes fewer, depending on the complexity of the chunks, the demands of the task, and the individual's current state of fatigue or distraction. 4 items. That is the working surface of the mind. The rest of what you know, the rest of what you have experienced, the rest of what you are, it sits in long-term memory, outside of conscious access, waiting to be retrieved when needed. This small capacity is not a bug. It is a carefully constrained design. A larger working memory would be slower, more metabolically expensive, and harder to manage. The brain is optimized for speed and efficiency at the cost of breadth. The scratchpad is small, but it is fast. And it has the entirety of long-term memory to draw from when it needs more. Short-term memory is fragile and brief. Without active rehearsal or emotional reinforcement, new information fades within seconds to minutes. Working memory and short-term memory are closely related, sometimes used interchangeably, though they refer to slightly different aspects of the same broad system. And both share a critical property: they are fragile, temporary, subject to rapid decay if not actively maintained. Think of the last time someone told you a phone number and you didn't write it down. Perhaps you repeated it to yourself, holding it in mind, actively rehearsing it, looping it through your awareness, saying it silently over and over to keep it alive. And if something interrupted that rehearsal, if someone spoke to you, or a thought arrived, or your attention was pulled elsewhere for even a moment, the number was gone. Completely and irretrievably gone, as though it had never been there. That experience of rapid, total forgetting is the natural behavior of short-term memory. Without rehearsal, without the active effort of holding information in the loop of conscious attention, it decays in a matter of seconds. The trace simply fades. No consolidation, no protein synthesis, no synaptic remodeling. The experience passes through without leaving anything behind. This is why encoding into long-term memory requires something more than mere exposure. Exposure alone, without attention, without emotional salience, without some form of repetition or elaboration, produces almost nothing durable. The brain needs a reason to invest the resources of consolidation. And fragile, unrehearsed short-term memory is the brain's way of saying, "This hasn't earned it yet. Show me why this matters, and then we'll talk about keeping it." Working memory is maintained by persistent neural firing, neurons that literally keep firing, continuously, to hold information in mind during the seconds it is needed. So how does working memory work, at the neural level? How does the brain hold something in mind, keep it present, keep it accessible, for the duration of a thought or a task? The answer, in its essential form, is surprisingly direct. The neurons that represent that information simply keep firing. They don't fire once and then pass the signal on. They fire repeatedly, continuously, maintaining an elevated level of activity that keeps the representation of the information alive in the neural network. This persistent firing is metabolically expensive. It consumes energy. It is, in a very real sense, an active holding operation, the brain working continuously, in real time, to prevent the information from decaying. The moment that persistent firing stops, the moment the neurons return to their baseline activity, the information is gone. This is why distraction is so devastating to working memory. When attention is pulled away, the persistent firing that maintains the current contents of working memory is disrupted. The neurons return to baseline. The information dissolves. And when attention returns, the scratch pad is blank. It is also part of why working memory degrades with fatigue and stress. Persistent neural firing requires metabolic resources, and when those resources are depleted, when the brain is tired or overwhelmed, the capacity to maintain the firing diminishes. The scratch pad gets smaller. Things fall off the edges more easily. The mental juggling act becomes harder and harder to sustain. The brain prioritizes novelty for storage. New, unexpected information is automatically flagged as more worthy of memory than information that is familiar and predictable. Your brain is, among other things, a prediction machine. Moment by moment, it is generating expectations about what is about to happen, based on past experience, on context, on the patterns it has learned. And it is continuously comparing what it predicted with what actually occurred. When reality matches the prediction, when what happens is exactly what the brain expected, very little special processing is triggered. The experience confirms what was already known. There is nothing to update, nothing to flag, nothing that requires extra resources. It passes through with minimal trace. But when something happens that violates the prediction, when something is new, or unexpected, or surprising, a very different response occurs. The mismatch between expectation and reality generates a strong signal, and that signal triggers increased attention, increased arousal, and, critically, increased encoding. Novel information is automatically prioritized. The brain, in effect, flags it unexpected, potentially important, worth remembering. This is why novelty is such a powerful memory aid. It's not simply that new experiences are interesting, it's that the brain is evolutionarily designed to treat unexpectedness as a signal of potential significance. In the ancestral environments in which human memory evolved, the unexpected was often dangerous, or valuable, or worth understanding. The brain learned, over millions of years, to pay extra attention to what it didn't already know. Novelty triggers the release of dopamine in the hippocampus, which acts as a store of this signal, essentially flipping a neurochemical switch that tells the brain to begin consolidating the experience. We can be more specific about how novelty prioritizes memory. The brain doesn't just detect novelty abstractly, it responds to it with a specific neurochemical event that directly influences the encoding process. When something novel is encountered, neurons in a region called the ventral tegmental area, part of the brain's dopamine system, deep in the midbrain, release dopamine into the hippocampus. And dopamine, in the hippocampus, acts as a powerful modulator of the synaptic plasticity that underlies memory formation. It enhances long-term potentiation. It makes the synapses that are currently active more susceptible to strengthening. It amplifies the encoding signal. In a very real sense, dopamine in the hippocampus functions as a store of this signal. A neurochemical instruction to the memory system: what just happened is worth encoding. Begin the process. This is part of why learning in a context of genuine curiosity and discovery tends to be more effective than learning in a context of repetitive, predictable drill. Curiosity generates novelty signals. Novelty signals generate dopamine. Dopamine enhances encoding. The brain is, at a neurochemical level, rewarding the exploration of what it doesn't already know. It is built to find the new world interesting, and built to remember it better for having found it so. Memory storage is guided by prediction errors. The gap between what was expected and what actually happened is a primary signal the brain uses to decide what needs to be learned and stored. We've been circling around this idea, and now we can name it directly. The signal that most powerfully drives learning and memory is not simply novelty in isolation, it is what neuroscientists call a prediction error. The measurable, calculable gap between what the brain predicted and what it actually experienced. When the prediction error is zero, when reality matches expectation perfectly, there is nothing to learn. The model of the world is accurate. No update is needed. When the prediction error is large, when something happens that was not predicted, or when something predicted failed to happen, the brain has received important new information. Its model of the world is wrong in some way, or incomplete. And that gap is the signal that triggers learning. Update the model. Adjust the weights. Store the evidence that led to the error so that the prediction can be improved next time. This computational framework, learning from prediction errors, is one of the most powerful ideas in both neuroscience and artificial intelligence. The brain, it appears, is not a passive recorder of experience. It is an active modeler of the world, continuously testing its predictions and using the errors those predictions produce to refine itself. Every surprise you have ever experienced has been, at the neural level, a learning opportunity. A signal that the map is not quite matching the territory. And your brain, patient and tireless, has been updating the map ever since. Emotional memories are stronger, more vivid, and longer lasting than emotionally neutral ones, and they remain more accurate over time in their core details. We touched on emotional memory earlier in this segment, but it is worth returning to with more depth. Because the effect of emotion on memory is not subtle, it is one of the most pronounced and consistent findings in all of memory research. Emotional memories are remembered better. They are retained longer. They are more resistant to the normal fading that erases most of our experience over time. And they tend to preserve their core content, the central, emotionally significant details, with greater fidelity than neutral memories preserve equivalent information. The mechanism involves the amygdala, as we've discussed. But it also involves the hormones released during emotional arousal, adrenaline, cortisol, norepinephrine, which act directly on the hippocampus and other memory structures to enhance consolidation. Emotional arousal is, neurologically, a multi-channel amplification of the encoding and consolidation processes. More resources are allocated. More redundancy is built in. The consolidation runs deeper and faster. There is a name for the particularly vivid, detailed memories that people form around highly significant emotional events, flashbulb memories. The moment you heard something that changed your life. The moment of sudden loss, or sudden joy, or sudden shock. These memories feel like photographs, bright, sharp, seemingly fixed. Research suggests that while they are not perfectly accurate, the peripheral details can be wrong, their core content tends to be preserved with unusual reliability. The emotional center of the experience survives intact. Stress hormones, particularly cortisol and adrenaline, directly shape how strongly memories are stored, with moderate stress enhancing consolidation and extreme stress potentially impairing it. Stress and memory have a complicated relationship. And that complexity is worth understanding clearly, because it is counterintuitive in parts. Moderate stress, the kind of mild arousal that comes with something that matters, something that requires your full engagement, enhances memory. The stress hormones released in moderate arousal, cortisol and adrenaline in particular, act on the hippocampus and amygdala in ways that boost LTP, enhance the consolidation of new memories, and increase the resources the brain dedicates to storing the experience. A mild challenge, a moderate emotional engagement, a situation that requires your attention and carries some consequence, these are, from a memory perspective, favorable conditions. The brain takes note. But extreme stress, the kind of overwhelming, uncontrollable, potentially traumatic stress that activates the body's full emergency response, has a different effect. Very high levels of cortisol, sustained for extended periods, can actually impair hippocampal function. The hippocampus is particularly sensitive to cortisol toxicity, and prolonged exposure can disrupt the very mechanisms that make memory formation possible. This is part of why traumatic events can produce fragmented, disorganized memories, intense in emotional quality but disordered in narrative structure. The amygdala may be highly activated, encoding the emotional signature of the experience with great power. But the hippocampus, overwhelmed and impaired, may fail to properly encode the contextual, sequential, narrative details. The result is a memory that feels vivid and present, but is difficult to place in time, difficult to narrate coherently, difficult to integrate into the ordinary flow of autobiographical memory. The stress response, in other words, was designed for moderate challenges, the kind that benefit from quick, sharp encoding of relevant details. It was not designed for the kind of prolonged, overwhelming horror that sometimes befalls human lives. And the memory system, sophisticated as it is, shows it seems most clearly at those outer extremes. We have arrived at the end of our fifth segment. And this one, perhaps more than any before it, has been about the recognizably human dimensions of memory. About attention as a gatekeeper. About emotion as an amplifier. About the tiny scratch pad of working memory and the fragile, flickering flame of short-term storage. About novelty and surprise and prediction errors. About the brain as something that is always, in a quiet and relentless way, trying to understand the world it finds itself in. And about the complex, nuanced, sometimes painful relationship between stress and the memories it leaves. You carry all of this with you. You have always carried it, without knowing the names for any of it. Every memory you have was shaped by these forces, by where your attention was, by what you felt, by what surprised you, by how safe or unsafe you felt in the moment of its formation. And knowing that doesn't change what you've lived. But it might, gently, change how you understand it. Let these ideas settle now. Let them find whatever place in you they're going to find. You don't need to do anything with them tonight. The brain is already handling it. Rest now. Welcome back to the quiet. The night is still here. Patient and unhurried, just as it always is. And so are we. In our last segment, we crossed into recognizably human territory, the gatekeeper role of attention, the amplifying power of emotion, the tiny fragile flame of working memory, and the complicated relationship between stress and the memories it leaves behind. We ended with something that might have lingered a little, the idea that every memory you carry was shaped by forces you didn't know had names. By the direction of your attention. By the weight of what you felt. By the neurochemistry of surprise. Tonight we continue in that same spirit. We begin with the darker edges of what stress and emotion can do to memory, not just strengthen it, but distort it, reshape it, lock it into patterns that can be difficult to escape. And then, midway through, we are going to make a turn that might feel, if you're still awake enough to notice it, genuinely wondrous. We are going to discover that the brain contains something like an internal GPS, a system for mapping the world in space and time that is so elegant, so unexpected, that its discovery earned a Nobel Prize. But we'll get there gently. There's no rush. Let's begin. Stress hormones can distort the content of memories, not just strengthen or weaken them, but actively shape what is encoded and what is left out. We ended our last segment with the observation that stress hormones have a complicated relationship with memory, that moderate stress enhances encoding while extreme stress can impair it. Tonight we go a step further, because the story is more nuanced still. Stress hormones don't just turn the volume of memory up or down. They change what the memory system pays attention to. And in doing so, they shape the content of what gets encoded, sometimes in ways that can mislead. Under stress, the brain's attention narrows. This is adaptive in a genuine emergency. You don't need to notice the color of the curtains when there is something dangerous in the room. You need to notice the threat, register its location and nature, and act. So the stress response prioritizes central, threat-relevant information at the expense of peripheral, contextual detail. This narrowing has a well-documented consequence in memory research, what is called the weapon focus effect, among others. In studies of eyewitness memory, people who witnessed a scene involving a weapon tended to remember the weapon itself with striking clarity, but remembered peripheral details, including the appearance of the person holding it, with significantly less accuracy than people who witnessed a comparable scene without a weapon. The stress response directed encoding resources toward the most salient threat-relevant element, and away from everything else. The memory feels vivid and confident, but it is selectively encoded, rich in some dimensions, impoverished in others. Stress hormones have not simply recorded the scene. They have edited it, before it was ever written down. Emotional arousal can trigger a process called synaptic hypertagging, where an emotional event retroactively strengthens the memory of neutral events that happen nearby in time. Here is something that might feel, at first, almost counterintuitive. We know that emotional events are powerfully encoded. But the reach of that emotional encoding extends beyond the emotional event itself. It can retroactively strengthen the memory of things that happen nearby in time, even if those things were completely ordinary and emotionally neutral on their own. The mechanism is related to the synaptic tagging we discussed in an earlier segment. When an emotional event occurs and triggers a surge of neuromodulators, dopamine, norepinephrine, the arousal-related chemicals, those neuromodulators don't affect only the synapses that were active at the moment of the emotional event. They flood more broadly through the relevant brain regions, and in doing so, they can capture the synaptic tags that were raised by recent, nearby activity associated with whatever was happening just before the emotional event. In practical terms, this means that a moderately encoded neutral memory, something you experienced shortly before an emotional event, can be retroactively upgraded. The emotional event, arriving afterward, provides the consolidation signal that the neutral event couldn't generate on its own. The neutral memory gets swept up in the emotional aftermath and stored more durably than it otherwise would have been. This is thought to be part of why people can remember, in unusual detail, the ordinary things they were doing immediately before a major shocking event. What you were eating, what song was playing, the entirely unremarkable conversation you were having, all of it, swept into durability by the emotional wave that followed. The brain's memory system is not just recording the present. It is, in a sense, casting backward in time, using the significance of what just happened to elevate the status of what came just before. The human memory system is evolutionarily tuned for survival. It is biased, at every level, toward encoding threats, losses, and negative outcomes more deeply than neutral or positive ones. There is a pattern in memory that almost everyone recognizes from their own experience, even if they've never had a name for it. Negative experiences tend to stick more readily than positive ones. A single harsh criticism can linger for days while a compliment evaporates almost immediately. One bad event in an otherwise good day can color the memory of the entire day. The embarrassing moment from years ago revisits you uninvited, while the many ordinary pleasant moments of those same years have become largely indistinguishable from one another. This is not a personal failing. It is biology. And it has a name: negativity bias. The memory system, shaped over millions of years of evolution in environments where threats were frequent and potentially lethal, is calibrated to prioritize negative information. A missed opportunity for food was unfortunate. A missed warning sign of a predator was potentially fatal. The asymmetry of consequences meant that the cost of failing to encode a threat was much higher than the cost of failing to encode a reward. And so the brain evolved to give extra weight to the negative, to flag it more urgently, encode it more deeply, and return to it more readily. The amygdala responds more strongly to threat than to reward. Cortisol and adrenaline, the stress hormones that amplify consolidation, are released far more readily by threatening stimuli than by pleasant ones. The whole system is tilted, from the ground up, toward remembering what went wrong. Understanding this doesn't make the bias disappear, but it can change the relationship you have with it. The harshness of your own memory is not a reflection of the true balance of your life. It is a reflection of what your ancient brain was built to prioritize. Your experience has almost certainly contained more light than your memory reports. The memory was just not designed to give light equal weight. Traumatic memories are stored differently from ordinary memories. They often bypass the hippocampal narrative encoding pathway and are stored in a more fragmented, sensory, and emotionally raw form. When an experience is overwhelming, when stress exceeds what the encoding systems can coherently handle, the normal pathway of memory formation is disrupted. And what results is a memory that feels fundamentally different from ordinary autobiographical recollection. Traumatic memories, as many people who have experienced them know, do not feel like stories. They do not have the organized, beginning, middle, and narrative structure of ordinary episodic memory. They feel more like fragments, sensory impressions, flashes of image or sound or physical sensation, divorced from their temporal context. They can arrive without warning, triggered by cues that may seem unrelated to the original event. And when they arrive, they can feel not like remembering something from the past, but like being suddenly, fully, back inside it. The neuroscience offers some insight into why. Under extreme stress, the prefrontal cortex, which normally helps organize and contextualize memory, providing the narrative frame, may be partially shut down by high cortisol levels. The hippocampus, similarly overwhelmed, may fail to properly encode the temporal and contextual scaffolding that allows a memory to be placed firmly in the past. But the amygdala, which responds to emotional and threat-relevant information, remains highly activated and encodes the emotional and sensory core of the experience with great power. The result is a memory that is emotionally and sensorially intense, but narratively incomplete. Rich in the feeling of the event, but poor in the contextual frame that would allow the brain to file it away as something that happened then, not something that is happening now. Understanding this is not a solution to trauma. But it is, perhaps, a form of compassion, a way of understanding why these memories behave as they do, and why the brain that holds them is not broken, but is doing the best it can with an experience that exceeded the system's design parameters. The sense of smell has a uniquely direct pathway to the brain's memory and emotion centers, which is why scents can trigger memories faster, more vividly, and more emotionally than any other sensory input. And now, something gentler. Something that almost everyone has experienced and wondered about. You walk past a bakery, or open a drawer that hasn't been opened in years, or catch a particular combination of cut grass and warm pavement, and something happens. Before you've had time to think, before any conscious process of recollection has begun, you are somewhere else. A specific kitchen. A specific afternoon. A specific feeling that you haven't had access to in years, suddenly present with an immediacy that almost takes your breath away. This is the Proustian memory, named for the French novelist Marcel Proust, who described it with extraordinary precision in his great work on time and remembrance, the moment triggered by the taste of a madeleine dipped in tea. And it happens because the olfactory system, the sense of smell, has a relationship with the brain's memory and emotion centers that no other sense can match. Every other sensory modality, sight, sound, touch, taste, routes its signals through the thalamus before reaching the higher cortical areas. The thalamus is a relay station, a processing hub that organizes and distributes sensory information. But olfactory signals bypass the thalamus entirely. They travel directly, with just one or two synapses, to the olfactory cortex, the amygdala, and the hippocampus, the very structures most directly involved in emotional memory. The directness of this pathway gives smell its extraordinary access to memory and emotion. The smell arrives at the memory center before the conscious mind has had time to form an interpretation. It is the most ancient of the senses, in evolutionary terms, and it has the most ancient and direct connection to the parts of the brain that remember and feel. No other sense reaches quite so far back, quite so fast. Memory storage is profoundly influenced by context. Memories are easier to retrieve when you are in the same physical, emotional, or physiological state you were in when they were formed. Here is a principle that has been demonstrated in some wonderfully unexpected experimental settings, and that most people have experienced without having a name for it. Memory is context-dependent. What you encoded in one context is most easily retrieved in the same context. The environment in which you learned something, the emotional state you were in, the physiological state of your body, all of these become woven into the memory trace itself, functioning as retrieval cues. When you return to a matching context, the cues activate the memory network, making recall easier and more vivid. In one of the more memorable demonstrations of this phenomenon, scuba divers were asked to learn lists of words either on land or underwater. They were then tested either in the same environment in which they learned, or in the other one. Words learned underwater were better recalled underwater. Words learned on land were better recalled on land. The environment had become part of the memory. Similar effects have been demonstrated for emotional states and even for pharmacological states. Information encoded while anxious is more easily retrieved when anxious again. Information encoded in one physiological state can be temporarily less accessible when the physiological state changes. This has a practical consequence that most students discover painfully at some point. Studying in a completely different environment from the one in which you'll be tested creates a context mismatch. The retrieval cues available in the exam room don't match those that were present during encoding, and access to the material is, as a result, somewhat impaired. But it also has a more tender implication. Returning to a place from your past, a childhood home, a street you used to walk, or room you haven't been in for years, can unlock memories that were apparently inaccessible, because the context itself is a key. The place remembers, in its way, what you encoded there. And being back in it gives you access to a version of yourself you thought you'd lost. Memory encoding is biased by expectation. The brain preferentially stores information that confirms existing beliefs, and sometimes distorts or dismisses information that contradicts them. We have established that the brain is a prediction machine, continuously generating expectations and comparing them with reality. In that prediction error, the gap between expectation and outcome is a primary signal for learning. Surprises are encoded with extra resources. What's expected passes through more quietly. But this system has a bias built into it. Because the brain's predictions are based on its existing model of the world, on what it already believes, information that fits the existing model tends to be processed more fluently, more easily, and with less friction than information that challenges it. And that processing advantage tends to produce a memory advantage. Information consistent with existing beliefs is encoded more readily, organized more efficiently, and retrieved more easily than information that contradicts those beliefs. Contradictory information is more likely to be dismissed, misremembered, or subtly distorted in the direction of the existing belief during the reconstruction that happens every time we remember. This is one of the deeper roots of confirmation bias, not merely a tendency to seek out confirming information, but a tendency to remember it more accurately and more durably. The brain, without any conscious intention, is running a slightly rigged filing system, one that is somewhat more receptive to what fits than to what challenges. Knowing this is important. Not because the bias can be simply switched off, it cannot, but because it suggests the value of deliberate, sustained exposure to information that challenges what we think we know. The brain will resist it. The encoding will be harder, the retrieval less smooth. But the effort of encoding something genuinely surprising, something that creates real friction with existing beliefs, is also, often, the effort that produces the most meaningful learning. The mind grows most at its edges. And now, having traveled through the darker and more complex territory of stress and trauma and bias, we arrive at something that feels, in contrast, like a kind of opening. An expansion. We turn toward space. The brain contains specialized neurons called place cells that fire only when you are in a specific location, creating a neural map of the space around you that is also the basis for spatial memory. In 1971, a neuroscientist named John O'Keefe made a discovery that would eventually earn him a share of the Nobel Prize in Physiology or Medicine. Working with rats navigating environments, he found something extraordinary: individual neurons in the hippocampus that would fire only when the animal was in a specific location in space. When the rat was anywhere else, the neuron was silent. When it returned to that particular spot, the neuron fired again. He called these place cells. And what they revealed was something that transformed the understanding of what the hippocampus actually does. The hippocampus, it became clear, is not simply a memory structure in the abstract sense. It is, among other things, a map maker. It contains a population of place cells that, together, create a neural representation of the space an animal inhabits, a kind of internal map, drawn not in ink but in patterns of selective neural firing, that tracks where the animal is, where it has been, and how the different locations of its environment relate to one another. This internal map is not just a convenience for navigation. It appears to be one of the fundamental organizing frameworks within which episodic memories of specific events, are encoded and stored. Events are not remembered in a spatial vacuum. They are remembered as having happened somewhere. And the place cell system provides the spatial coordinate within which the memory is anchored. Where you were is part of what you remember. And the brain has dedicated neurons whose entire purpose is to know exactly where that was. Place cells create what are called place fields, specific zones in physical space that each neuron responds to, and together they tile the entire environment with overlapping coverage. A place cell doesn't respond to a single point in space, a single precise coordinate, like a pin in a map. It responds to a region, an area of space, typically ranging from a few centimeters to a meter or more across, depending on the size of the environment within which the cell's firing rate is elevated. Outside of this region, the cell is largely silent. This responsive zone is called the place field. Now, in any given environment, many place cells are active simultaneously, each with its own place field centered on a different location. Together, these overlapping fields tile the entire environment. Every part of the space is represented by at least one, usually several, active place cells. The population of active cells, at any moment, provides a complete and continuous representation of the animal's current position. What is particularly elegant is that this map is self-organizing. The place fields don't need to be pre-specified or programmed. When an animal enters a new environment for the first time, the place cell system rapidly assembles a new map, distributing the fields across the novel space, creating a unique representation of that particular environment within minutes. The hippocampus is not consulting a pre-existing map of the world. It is generating a new one, on the fly, every time it encounters a new space. And this newly generated map immediately becomes available as a spatial scaffold for encoding the events that happen within that environment. The map-making and the memory-making are, in the hippocampus, deeply intertwined, two aspects of the same fundamental capacity. The spatial tags provided by place cells help anchor memories to the locations where they were formed, which is part of why returning to a place can unlock memories that seemed inaccessible. We come full circle to something we touched on a few facts ago, the context dependence of memory. And now we can see the neural mechanism that underlies at least part of that phenomenon. When an event is encoded, the place cells that are active at the time of encoding, the cells whose place fields cover the location where the event occurs, are incorporated into the memory trace. They become part of the neural pattern that represents the memory. The place, in a very literal sense, becomes part of the memory. When you later return to that location, the same place cells are reactivated by the physical environment itself. The neural map is rebuilt, the same cells fire, and that reactivation serves as a retrieval cue. It partially reinstates the neural context in which the memory was formed, making the memory more accessible, more easily triggered. This is the neuroscience of the experience we described earlier, the unlocking of memory by returning to place. The place cells remember the location. And in remembering the location, they help reinstate the context of what happened there, reaching back through time to the experience that was encoded in that particular patch of space. It is, when you think about it, one of the most beautiful things the brain does. It builds a map of everywhere you have ever been. And in that map, woven into every location, are the threads of everything that happened to you there, waiting, patiently, for you to return and find them again. We have covered a great deal of terrain in this segment. We began in the difficult territory of stress distortion and traumatic memory, moved through the ancient power of smell, through the context dependence of recall, through the brain's built-in bias toward confirming what it already believes, and then arrived, finally, at something genuinely wondrous, a population of cells in your hippocampus that know exactly where you are, that map the world you move through, and that weave the geography of your life into the fabric of your memory. Your brain contains an internal map of everywhere you have ever been. Every room, every street, every landscape, every space that has mattered to you, it is in there, represented in the quiet, selective firing of cells you will never be able to feel working. And in every location on that map, something is stored. Some fragment of what it felt like to be you, in that place, at that time. Let that settle. Let the map be as large as it is. And let yourself rest now, in whatever place you are, knowing that this too is being added to it. Sleep well. Welcome back to the quiet. The night continues, unhurried and generous, as it always is. And wherever you are right now, fully awake and listening carefully, or drifting at the soft edge of sleep where the voice becomes something more like a current than a conversation, you are exactly where you should be. We have come a long way together through this landscape of memory. We've looked at the molecular machinery, the neural circuits, the role of sleep, the power of emotion, the map-making place cells of the hippocampus. We've descended to the level of calcium ions and epigenetic marks, and pulled back to the scale of the whole distributed brain, engram cells spread like constellations across its regions. Tonight we begin somewhere perhaps a little unexpected, with time. With the question of not just where memories are anchored in space, but when they are anchored in time. And then, midway through, we are going to venture into territory that is psychologically fascinating, and perhaps a little humbling. Because we are going to discover that memory is not just fallible. It is actively creative. And sometimes, what it creates is something that never happened at all, but will arrive there gently. Let's start at the beginning of the segment, in the brain's relationship with time. The brain tags memories not only by their content and location, but by when they happened, and dedicated neural machinery tracks temporal context during encoding. We know that memory is contextual, that where you were when something happened becomes woven into the memory trace, and that returning to the place can help unlock the memory. But space is only one dimension of context. The other is time. And the brain is as attentive to when as it is to where. Every memory you form is tagged, in some fashion, with temporal information. Not with a precise timestamp. Memory is not a clock, and it is notoriously poor at accurately locating events in absolute time. But with relative temporal context. The sense of before and after. The understanding of which memories are close together in time and which are far apart. The feeling, when you retrieve a memory, of whether it is recent or distant, whether it belongs to last year or to childhood. Some of the same hippocampal circuits involved in spatial mapping appear to also handle temporal mapping. There are neurons, sometimes called time cells, that fire at specific moments within a sequence of events, creating a kind of temporal scaffold, a neural record not just of what happened but of the order in which it happened and the duration over which it unfolded. This temporal tagging is essential for episodic memory, for the specifically human capacity to mentally time travel, to place yourself back in a specific moment and re-experience it as a located event in your personal history. Without the temporal tag, a memory might feel more like a fact, something you know happened, but not something you can inhabit, not something with a before and an after and a particular quality of that specific time. The one of memory is as real a part of the memory as the what. And the brain keeps careful, if imperfect, track of both. The brain uses event boundaries to chunk continuous experience into discrete, memorable episodes, each boundary acting as a kind of mental paragraph break in the ongoing story of your life. Life, as it is actually lived, does not arrive in neatly separated episodes. It is a continuous, unbroken stream of experience, one moment flowing into the next without gaps or titles or intermission. And yet memory does not store it that way. Memory stores it in chunks. In episodes. In units that have something like a beginning and an end. The mechanism behind this chunking involves what cognitive neuroscientists call event boundaries, the moments when something in the situation changes significantly enough to signal a transition from one episode to the next. A change in location. A change in social context, when someone leaves or arrives. A change in the goal or activity you are engaged in. A significant shift in what is happening. At event boundaries, the hippocampus appears to do something special. It processes the transition, closes off the current episode, and begins a new encoding context for what comes next. Like a writer who hits return and begins a new paragraph. The continuous stream of experience is broken, at these natural joints, into discrete and separable units. This chunking has practical consequences for how we remember. Events that happen within a single episode tend to be strongly associated with one another, linked in memory, easy to retrieve together, difficult to separate. Events that happen across an episode boundary, even if they were close together in clock time, tend to feel more temporally distant from one another, more difficult to access as a pair. the boundary creates a kind of temporal gap in memory that is larger than the actual gap in time. This is part of why days that were full of varied activity and transitions feel, in memory, longer and richer than days spent in a single unchanging environment doing one thing. More event boundaries means more episodes, means a richer and more expansive subjective sense of time having passed. Strong event boundaries, significant transitions that signal a major change in context, are particularly effective at creating distinct, durable new memories. Not all event boundaries are created equal. The transition from one room to another is an event boundary, but a subtle one, a gentle paragraph break in the story. The transition from one chapter of your life to another, a move to a new city, the beginning or ending of a significant relationship, the first day of something that matters, is a boundary of a different order altogether. Strong event boundaries, marked by significant changes in context, goals, environment, or emotional state, create particularly clear demarcations in memory. They serve as powerful anchors around which new memories can organize themselves. The period after a major transition tends to be remembered differently from the period before it. It feels like a different chapter, because, in the architecture of memory, it largely is. Strong event boundaries also appear to benefit from a kind of reset in the encoding system, a clearing of the short-term context that was accumulating, a fresh start in which new information is not confused with what came just before. The boundary creates the conditions for a new episode to begin cleanly, without the interference of the previous one. This is one of the reasons why significant life transitions, even difficult ones, can paradoxically be accompanied by a sharpness and clarity of memory that ordinary periods lack. The strong boundary tells the encoding system, something new is beginning here. Pay attention. Start fresh. And the memory system, responding to that signal, does exactly that. Some memories are stored in parallel branches, separate neural representations of the same event that encode different aspects or interpretations simultaneously. Here is something that might surprise you. When something happens to you, the brain does not necessarily store a single unified version of the event. In some cases, particularly for experiences that are ambiguous, emotionally complex, or rich with different kinds of information, multiple parallel representations may be formed, separate neural traces that encode different aspects or framings of the same event at the same time. Think of a conversation that was important to you, one that might have been interpreted in several ways, or that carried both warmth and tension simultaneously. It is possible that your brain stored not one version of that conversation but something more like a family of related traces, one that emphasizes the emotional texture, one that encodes the factual content, one that holds the spatial and sensory context, each in its own circuits, each available as a somewhat different angle on the same experience. These parallel branches are not experienced as multiple separate memories. When you recall the event, what you experience feels unified, assembled from whichever branches are most activated by the current retrieval cue. But the underlying representation may be more multiple, more branching, than the experience of remembering suggests. This parallel architecture is part of what makes memory both flexible and vulnerable. Flexible, because different retrieval cues can access different branches, allowing the same event to be remembered from different angles depending on what prompts the recall. Vulnerable, because different branches can drift apart over time, influenced by different subsequent experiences, leading to a gradual divergence in the parallel representations of the same original event. Memory traces from different experiences overlap in the neural circuits that store them, sharing some of the same neurons and synapses, but the brain maintains their distinctiveness through subtle differences in the pattern. The brain has a limited number of neurons, and it has a vast number of memories to store. These two facts inevitably lead to overlap, to situations where the neural circuits that encode different memories share some of the same cells. This overlap is not a flaw to be engineered away. It is a feature that enables generalization. When memories share neural real estate, the patterns that are common across similar experiences are naturally reinforced, while the patterns that are unique to individual experiences are preserved in the specific details of which neurons are active and how strongly. The overlap is the mechanism by which the brain abstracts from the particular to the general, by which it learns that this thing and this other thing are both instances of a broader category. But the overlap also creates risk. Similar memories, stored in overlapping circuits, can interfere with one another. Details from one event can bleed into another. The specific features of two similar conversations, two similar holidays, two similar faces, can become confused, the memories drifting into one another through their shared neural substrate. The brain counteracts this through a process called pattern separation, the ability to make similar inputs less similar in their neural representation, sharpening the distinctions between memories that would otherwise risk merging. The dentate gyrus, a subregion of the hippocampus, appears to be particularly important for this pattern separation function, helping maintain the distinctiveness of similar memories even as they share overlapping neural resources. It is a continuous, invisible balancing act between the need to generalize and the need to distinguish, between the usefulness of finding patterns and the necessity of remembering individuals. Memory encoding is scaffolded by familiar routines. Regular, predictable patterns of experience create a structural framework within which new and distinctive memories can be more easily organized and retrieved. Routines, it might seem, should be the enemy of memorable experience. If every morning is more or less like every other morning, how does any particular morning stand out? And there is truth in that. The details of individual routine mornings tend to blur together, not because nothing happened, but because nothing departed sufficiently from the familiar script to trigger strong event boundaries or generate significant prediction errors. The brain, recognizing the familiar pattern, processes it efficiently and economically, without investing the extra encoding resources it reserves for the novel and unexpected. But routine also plays a positive role in memory, not by making individual instances memorable, but by providing a stable scaffold against which departures from routine stand out with particular clarity. Because the routine is so well encoded and so automatically processed, anything that deviates from it is immediately flagged as a prediction error. It registers as surprising, as worthy of attention, as something that doesn't fit the usual pattern and therefore deserves extra encoding resources. The familiar routine is, in this sense, a baseline that makes the unusual legible. You remember vividly the morning something unexpected happened precisely because all the other mornings were predictable enough to fade into background. The scaffold of the ordinary is what makes the extraordinary stand out. Routine and novelty are not opposites in the architecture of memory. They are partners. The routine provides the structure that gives novelty its meaning. And now we arrive at a turn in our journey. We leave the architecture of encoding behind, and we enter perhaps the most psychologically intimate territory we have visited yet. We are going to talk about what happens to memories after they are formed, about how they change, about the ways the brain, in the act of remembering, sometimes quietly rewrites what it finds. Memories are not fixed at the moment of formation. They can be updated, revised, and changed throughout the course of a lifetime, with each act of recall potentially altering the stored trace. We touched on reconsolidation earlier in our journey, the discovery that when a memory is retrieved, it re-enters a briefly plastic, vulnerable state before being restabilized. Now let's sit with the full implications of that finding. Every time you remember something, you are doing more than accessing a fixed record. You are reconstructing the memory, reassembling it from its distributed components, drawing it back into active neural representation. And in that moment of active representation, the memory is, briefly, a living thing again. Capable of being influenced, capable of absorbing new information, capable of being changed. This means that the act of remembering is not neutral. Every time you recall a memory, you are also, in some small way, altering it. The new context in which you remember, your current emotional state, your current knowledge, the cues that prompted the recall, all of these leave traces on the memory as it is reconsolidated. The memory that is stored again after each retrieval is not quite the same as the memory that was stored before. Over a lifetime of remembering, over the many times a significant memory is returned to, thought about, talked about, revisited, the cumulative effect of these small alterations can be substantial. The memory of something that happened 30 years ago is not a pristine record of 30 years ago. It is a palimpsest, a document that has been written on again and again, each iteration leaving its own layer of revision over what came before. We remember the past, but we remember it from the present. And the present is always changing who we are. Memory can be actively rewritten during recall, through a process called reconsolidation. New information encountered during or after retrieval can be incorporated into the stored memory trace. We have named reconsolidation before. Now we can look at its implications clearly, because they are remarkable and sometimes unsettling. If a memory, when retrieved, enters a plastic state, if it becomes briefly open to modification before being restabilized, then it is possible, in principle, to deliberately modify a memory by introducing new information during or immediately after its retrieval. And experiments have demonstrated that this is not just possible in principle but happens readily, with relatively simple manipulations. In a classic series of experiments, people were shown footage of a car accident and later asked about it. When the question used the word smashed rather than hit to describe the collision, participants later reported having seen broken glass at the scene, even though no broken glass had been present in the footage. The language of the question, encountered after the memory was retrieved, had been incorporated into the stored memory itself. The memory was rewritten, subtly, by what came after. This has profound implications for the reliability of eyewitness testimony, for the way therapeutic conversations can influence the memories they explore, for the way the stories we tell about our own lives gradually reshape the memories those stories are based on. It also has an implication that is, in a different light, quietly hopeful. Because if memories can be rewritten, if the emotional valence, the narrative framing, the meaning of an experience can be altered through the process of reconsolidation, then it becomes possible, in principle, to revisit painful memories in contexts of safety and support, and to allow the restabilization to incorporate something new. Not to erase what happened, but to change the relationship between who you are now and what was stored then. The plasticity of memory is not only a vulnerability. It is also, sometimes, a path toward healing. False memories, vivid, detailed recollections of events that never actually occurred, can be created experimentally and are indistinguishable, to the person experiencing them, from genuine memories. This is perhaps the most unsettling thing we will encounter in our entire journey through the science of memory. And it deserves to be stated plainly. False memories are real. They can be created. And they feel exactly like genuine memories. The psychologist Elizabeth Loftus has spent decades demonstrating this with extraordinary precision. In one of her most famous paradigms, participants were given a booklet of childhood memories described as having been provided by their family members. Most of the memories were real. But one was invented, a detailed account of having been lost in a shopping mall as a child, a frightening experience that in fact had never happened. Over repeated interviews, a significant proportion of participants not only came to believe the invented event had occurred, they began to elaborate on it, adding sensory details, emotional nuances, specific memories of the people involved. They were not lying. They were not performing. They believed, fully and without reservation, that they were remembering something real. The false memory was phenomenologically indistinguishable from their genuine ones. This can happen because memory reconstruction draws on the same processes, the same sensory imagery, the same emotional systems, the same narrative construction, regardless of whether the source of the reconstruction is a genuine past experience or an imagined, suggested, or confabulated one. The brain doesn't stamp memories with a mark of authenticity. It assembles what it can from available sources, and the assembly, when complete, feels real. The implications ripple outward into law, into therapy, into the stories we tell about ourselves and about others. They are not comfortable implications, but they are true. And knowing them is part of knowing what memory actually is. Memory is profoundly shaped by social and cultural context, what we remember, how we remember it, and even whether we remember it at all is influenced by the communities and narratives we belong to. We do not remember alone. Memory is, from the beginning and throughout, a social process. The stories we tell about our experiences, to friends, to family, to ourselves, shape the memories those stories are based on. The act of narrating an experience strengthens some aspects of it and suppresses others. Details that are included in the telling are reinforced, details left out are allowed to fade. Over many tellings, the story and the memory gradually converge, until the memory of the experience has become, in significant part, the memory of the story. And the stories we tell are shaped by the social and cultural contexts we inhabit. What is considered worth remembering? What narrative frames are available for making sense of experience? What emotions are considered appropriate to express and therefore to encode? All of these are culturally mediated. Different cultures organize autobiographical memory differently. Different families have different collective narratives into which individual memories are fitted. Different historical moments make certain stories tellable and others not. Memory, in this deepest sense, is not a private, individual faculty. It is a communal one. We remember together. We remember through the frameworks that our communities provide. We remember in the language we share, using the narrative structures our culture has made available. And what we cannot share, what has no language, no framework, no social space, is, for many people, genuinely harder to hold. This is not an argument for the unreliability of memory. It is an argument for understanding memory as something embedded in relationship and community, shaped by connection, by shared narrative, by the particular human world in which each of us finds ourselves remembering. We are not the sole authors of our memories. We write them, always, in a language we learn from others. And we tell them, always, to someone, even when that someone is only ourselves. And so we have reached the end of our 7th segment. We began with the brain's relationship to time, the temporal tags, the event boundaries, the chunking of continuous experience into discrete episodes. We moved through the parallel, overlapping, scaffold architecture of memory storage. And then we turned into perhaps the most psychologically intimate territory of this entire journey, the rewritability of memory, the reality of false memories, and the social nature of remembering. What has emerged, through all of this, is a portrait of memory that is very different from the recording device we might have imagined at the start. Memory is not a camera. It is not an archive. It is not a fixed record that can be trusted to be faithful. It is something stranger and richer than any of that. It is a living, continuously revised, socially embedded, biologically constrained act of reconstruction, carried out, moment by moment, by a brain that is doing its absolute best to make sense of what has happened, what is happening, and what might happen next. And that brain, right now, in the quiet of wherever you are, is doing exactly that. Sorting and revising and reconstructing. Making sense of another day. You can let it work without your help. That's what nights are for. Rest now. Welcome back to the stillness. The night is deep now. And wherever you are in your listening, whether this is your first segment or your 8th, whether you've been here since the beginning or you've arrived somewhere in the middle, the voice is the same. Unhurried. The same pace as breathing. The same patience as dark. In our last segment, we entered some of the more psychologically intimate territory in this entire journey. We discovered that memory is not fixed, that it is rewritten each time it is recalled, that false memories are indistinguishable from real ones, that what we remember is shaped by the social and cultural worlds we inhabit. We left with something that might have felt a little destabilizing, the recognition that the record of our lives is more fluid, more constructed, more collaborative than we typically assume. Tonight, we are going to approach memory from a different angle. We are going to think about it not just as a psychological phenomenon, not just as a biological mechanism, but as something with a kind of physics to it. Something with energy costs and efficiency pressures and architectural constraints. Something that has been shaped over millions of years of evolution, not just to remember well, but to forget well. To manage its own resources. To stay lean and functional in a biological system that cannot afford to be profligate. Because forgetting, it turns out, is not simply failure. Forgetting is, in many cases, the point. Let's begin. Memory traces are not fixed readouts, they are readout sensitive, meaning the way a memory is retrieved changes depending on the cues available, the current context, and the state of the network at the moment of recall. We have established, over many segments now, that memory is reconstructive rather than reproductive, that remembering is an act of assembly rather than playback. But let's go one step further, because there is something even more fundamental to say about the nature of memory retrieval. A memory trace is not a fixed message waiting passively to be read. It is a potential, a pattern of synaptic weights and engram cell connections that can be activated, partially or fully, by the right combination of retrieval cues. And the retrieval is always, in a meaningful sense, a negotiation between the stored pattern and the current state of the system attempting to retrieve it. Different cues activate different aspects of the stored pattern. Different emotional or physiological states at the moment of retrieval make different parts of the network more or less accessible. Different amounts of time since encoding change how the trace responds to activation. The same underlying memory can produce very different retrieved experiences depending on the circumstances of the retrieval. This is part of why memory feels different on different days. Why something you struggle to recall on one occasion surfaces effortlessly on another. Why a memory can feel distant and factual one day and painfully present the next, not because the stored trace has changed dramatically, but because the conditions of retrieval have changed. The cues available. The emotional resonance. The neural state of the system doing the remembering. The trace is consistent. But the readout is always in conversation with the present moment. Some memories are stored but effectively silent. They exist as latent patterns in neural circuits, invisible to ordinary conscious recall, until a specific trigger reactivates them. This is one of the quieter but more remarkable things the science of memory has revealed. Not all stored memories are equally accessible under ordinary circumstances. Some are, in a functional sense, silent, present in the structure of the neural network, encoded in the pattern of synaptic weights, but not activated by normal retrieval attempts. They are there, but they are not answering. These silent memories can remain inaccessible for years, sometimes for the duration of a lifetime. But they are not gone. The right trigger, a specific sensory cue, an unexpected context, a particular emotional state, can reach into the network and suddenly reactivate a pattern that had been dormant for decades. This is the experience many people have of encountering a smell, or a piece of music, or a particular quality of light, and being overtaken by something they did not know they still remembered. The memory was not retrieved before because the ordinary retrieval system had no route to it, no accessible cue, no context that matched the encoding conditions well enough to activate the pattern. But the pattern was there all along, intact and waiting. There are also, in the research literature, more dramatic examples, cases of deeply encoded early memories, formed before the age of conscious narrative recall, that can be triggered by specific circumstances even when they cannot be deliberately accessed. The brain holds more than we can find when we look for it. The archive is larger than the index. Memory circuits that are frequently activated remain strong, accessible, and well-maintained, while circuits that are rarely activated gradually weaken through a process of passive decay and active pruning. Use it or lose it is one of the most ancient and broadly applicable principles in biology. And it applies to memory with particular force. Memory circuits are not static structures that, once formed, simply persist unchanged unless something actively disrupts them. They require maintenance. The synaptic connections that constitute a memory need to be periodically reactivated, through recall, through related experience, through the replay that happens during sleep, in order to remain strong and accessible. Without that periodic reactivation, the trace weakens. The synaptic weights drift back toward baseline. The pattern becomes harder to reactivate, requiring stronger or more specific cues to trigger. And eventually, if the neglect is long enough, the trace may become effectively irrecoverable, not because the neurons themselves are gone, but because the specific pattern of connections that encoded the memory has faded below the threshold of practical retrieval. Conversely, memory circuits that are frequently activated are continuously maintained and reinforced. The memories you return to regularly, that you think about, talk about, revisit in imagination, are the memories that remain vivid, specific, and easily accessible. They are being reinforced, structurally, by each activation. Each recall is, in a small way, a fresh consolidation. This is a principle with both a reassuring and a slightly sobering implication. The memories you tend to carry with you are the ones you have, in part, chosen to carry. In the memories you have neglected, the experiences left unvisited, the details never rehearsed, are quietly, inevitably, fading. The brain is fundamentally wired to forget most of what it experiences. Forgetting is the default, and remembering is the exception that requires specific conditions to occur. This might be the most important refrain we offer in this entire series of segments. Because most of us, when we notice ourselves forgetting something, experience it as a failure. As something going wrong. As the memory system not doing its job. But the truth is precisely the opposite. Forgetting is the memory system doing its job. Remembering, in any durable form, is the exception. Forgetting is the rule. Of the continuous flood of sensory information and experience that passes through your brain each day, the faces glimpsed on a street, the words of 100 brief conversations, the texture of every meal, the precise sequence of every ordinary hour, the vast majority leaves no lasting trace. Not because the memory system failed to record it, but because the memory system made a decision, quickly and without your conscious input, that it was not worth the resources of long-term storage. This default toward forgetting is not a design flaw inherited from our evolutionary past. It is the design. A system that remembered everything equally would be unusable, overwhelmed by irrelevant detail, unable to prioritize, unable to generalize, unable to recognize that today's cup of coffee is relevantly similar to yesterday's even though no two cups are identical in every detail. Forgetting is what allows the memory system to be efficient, selective, and useful. It is the necessary counterpart to the selectivity that makes memory meaningful. You are not a failing remembering machine. You are a highly selective one. And the selection, most of the time, is being made wisely. The brain actively deletes weak memory traces, not through passive decay alone, but through dedicated molecular mechanisms that dismantle synaptic connections that are no longer being reinforced. We might imagine forgetting as purely passive, a slow fading, like a photograph left in sunlight, where the image dims simply through the absence of whatever keeps it vivid. And passive decay is real. Unreinforced memories do fade over time. But forgetting is not only passive. The brain also actively dismantles memories it has determined are no longer worth maintaining. There are dedicated molecular mechanisms, proteins and signaling cascades, whose specific function is to depress synaptic strength, to undo some of what LTP has built, to prune back connections that are not earning their metabolic keep. One of the key players in this active forgetting is a protein complex called the RAC1 pathway, involved in the degradation of the actin cytoskeleton that supports synaptic spine structure. When a synaptic connection is weakened or marked for removal, the structural scaffolding that supports it, the physical architecture of the dendritic spine, is actively dismantled. The synapse doesn't just fade. It is taken apart. This active pruning is most dramatic during certain developmental periods, the extensive synaptic pruning that occurs during adolescence, for example, when the brain removes a substantial proportion of its synaptic connections in a massive reorganization that shapes the adult neural architecture. But it continues, in a more measured way, throughout life. The brain is always editing, always revising, always removing what it has decided it no longer needs. Forgetting, in this light, is not entropy. It is curation. The brain forgets on purpose. Active forgetting mechanisms exist to reduce interference between similar memories and to keep the memory system functional and precise. The case for purposeful forgetting becomes even clearer when we consider what happens when forgetting fails. There are rare individuals who appear to be unable to forget, who retain, with almost perfect fidelity, the details of every day of their lives. This condition, sometimes called highly superior autobiographical memory, might sound like an extraordinary gift. And in some respects it is. These individuals can retrieve specific dates and events from their lives with astonishing precision. But they also report that living with this kind of memory is, in important ways, burdensome. The past is always present. Every day re-evokes memories of other days. The inability to let things fade means that old emotional experiences remain as vivid and as powerful as recent ones. The natural psychological process of moving on, of allowing the intensity of past experiences to diminish, of gaining the perspective that comes with distance, is compromised. And at the neural level, When all traces are retained with equal strength, a different problem emerges: interference. When many similar memories are all strongly encoded, they can interfere with one another during retrieval, making it harder, not easier, to access any specific one accurately. The precision of memory depends, paradoxically, on the willingness of the system to let most of it go. Forgetting is how the brain keeps the signal clean. How it maintains the resolution it needs to distinguish this specific Tuesday from all the other Tuesdays. How it prevents the past from drowning the present. It forgets, actively and purposefully, so that what remains can be clearly seen. The brain forgets to protect itself from overload. Without active clearing of no longer relevant information, the memory system would gradually become saturated and unable to incorporate new learning. There is also a more immediate, practical reason for forgetting. The synaptic resources of the brain, the connections available for encoding new memories, are finite. Not infinite, as we noted when considering storage capacity, but finite in a different sense. At any given moment, the number of synapses available to undergo LTP, to be strengthened by new experience, is limited by the current state of the network. If memories were never forgotten, if every encoded trace remained at full strength indefinitely, the network would gradually approach saturation. The signal-to-noise ratio would decline. New information would have difficulty finding strong, reliable encoding, because the synaptic landscape would already be densely occupied by the accumulated traces of the past. Forgetting, the weakening and removal of old, low-priority traces, continuously clears space. It restores the capacity for new encoding. It allows the system to remain sensitive to novelty, responsive to change, able to update its models of the world in light of new experience. Sleep appears to play a role in this clearing, the synaptic homeostasis process we described in an earlier segment, the gentle downscaling of overall synaptic strength that happens during slow-wave sleep, serving not only to consolidate important memories but to free up the system's capacity for tomorrow's learning. Every night, in the quiet of your sleeping brain, a kind of clearing happens. The housekeeping that makes tomorrow possible. The forgetting that makes tomorrow's remembering easier. Old memories are not stored in a fixed layer from the past, they are continuously, thinly updated over time as new related experiences are woven into the existing trace. Here is something that quietly unsettles our usual way of thinking about old memories. We tend to imagine them as artifacts, fixed things from the past that we access unchanged, like photographs in an album. The memory of my 5th birthday is the memory of my 5th birthday, unchanged and unchanging, waiting in its place. But we have already seen that every retrieval is a reconstruction. And we have seen that reconsolidation allows new information to be incorporated into existing traces. And so it should not entirely surprise us to learn that old memories are not quite the fixed artifacts we imagine. They are living traces, and they continue to receive thin updates as new related experiences are processed. When you encounter something today that is related to an old memory, a new experience that shares some features, a conversation that revisits familiar themes, a piece of information that adds to something you knew long ago, the new experience doesn't simply sit alongside the old memory in isolated parallel. It interacts with it. The old trace, activated as a context for the new experience, is also subtly updated by the encounter. The new information is threaded into the old pattern, enriching it, sometimes revising it, sometimes adding a new dimension to something that seemed complete. This is, in many ways, a beautiful thing. Our memories of the past continue to be enriched by the present. The person we are becoming continues to annotate the record of who we have been. Old experiences are continuously being seen through new eyes, not replaced, but illuminated differently by the accumulated light of everything that has happened since. The archive is not a museum. It is a living document. And it is always, in its quiet way, still being written. And now, in the final two facts of this segment, we turn towards something that grounds all of this in the deepest physical reality of the brain. We have spoken of memory in biological terms, in molecular terms, in systems-level terms. Now we consider it in terms of energy, of weight, of the fundamental physics of a biological machine doing one of the most complex things any physical system in the known universe has ever been asked to do. Memory storage is an energy-intensive process, the synthesis of new proteins, the remodeling of synapses, and the maintenance of memory traces all impose real metabolic costs on the brain. The brain is the most metabolically expensive organ in the human body. It constitutes roughly 2% of body weight but consumes roughly 20% of the body's total energy budget at rest. And a significant portion of that energy expenditure is dedicated to the processes of memory, to the synthesis of the proteins that remodel synapses, to the maintenance of the ionic gradients that allow neurons to fire, to the persistent neural activity that holds information in working memory, to the replay and reconsolidation that happens during sleep. Every memory you form has a real energy cost. The protein synthesis required for long-term storage requires metabolic resources. The remodeling of dendritic spines requires metabolic resources. The maintenance of strengthened synaptic connections against the natural tendency toward decay requires a continuous investment of metabolic resources. This energy cost is part of why the brain is so selective about what it stores. Encoding everything at the depth required for long-term memory would be metabolically unsustainable. The selectivity of memory, the gating function of attention, the filtering function of emotional salience, the active forgetting mechanisms we have been discussing, is not just computationally sensible. It is energetically necessary. The brain cannot afford to remember everything. And so it has evolved, over millions of years, a system of extraordinary discernment, one that allocates its metabolic investment in memory to the experiences that, by the available indicators, seem most worth remembering. Your memories have a cost. And the fact that they were kept means, in a real biological sense, that they were worth it. The memory system is designed for energy efficiency. It achieves its remarkable capacity and performance not by brute force, but through elegant architectural solutions that do more with less. And yet, for all the metabolic cost of memory, the brain is also, by any reasonable engineering standard, extraordinarily efficient. The computational power packed into the roughly 1.4 kilograms of tissue that constitutes a human brain, the capacity to store and retrieve vast amounts of information, to learn from experience, to generalize, to integrate, to imagine, vastly exceeds anything we have built with silicon at a fraction of the energy cost. A modern supercomputer might consume megawatts of power. The human brain runs on approximately 20 watts, roughly the power of a dim light bulb. And it does so while performing tasks that no supercomputer can yet match in flexibility, in contextual sensitivity, in the richness and nuance of what it handles. The efficiency comes from architecture. From the distributed, overlapping nature of memory storage, the fact that the same neurons and synapses serve multiple memories simultaneously, through subtle differences in pattern, rather than each memory requiring dedicated private hardware. From the sparse coding strategies that the brain uses, encoding memories in the pattern of activity across a large population of neurons, with each individual neuron being active in only a small fraction of all memories, minimizing the metabolic cost of any individual memory while allowing an astronomically large number of possible patterns. From the hierarchical organization of processing and storage that routes different kinds of information through specialized circuits rather than handling everything in one place. And from forgetting. Always, again, from forgetting, the elegant, purposeful, curatorial process that keeps the system lean, keeps the signal clear, keeps the capacity fresh. The brain is not a warehouse filling up. It is a living system, continuously managing its resources, continuously making choices about what to hold and what to release. It is, when you contemplate it clearly, one of the most extraordinary things that exists. 20 watts. 1.4 kilograms. And inside it, the accumulated weight of a human life. We have come to the end of our 8th segment. And this one has taken us somewhere perhaps unexpected, not into the richness and drama of memory, but into its physics. Its energy budget. Its architecture of efficiency. The beautiful paradox of a system that achieves its remarkable capacity not by brute force but by discernment, by selectivity, by the willingness to forget. Forgetting, we have seen, is not failure. It is design. It is curation. It is the thing that makes the memory system sustainable, precise, and alive. And right now, as the night continues its quiet work, your brain is engaged in exactly this, the careful, energetically costly, elegantly efficient management of everything you are. Consolidating what matters. Releasing what doesn't. Replaying the day in compressed, distributed form. Pruning and reinforcing. Clearing space. Making room. 20 watts of soft biological light, doing the most remarkable thing in the known universe. You don't need to do anything to help. You just need to rest. Let it work. Good night. Welcome back to the stillness. If you've been here since the beginning of this journey, you've now traveled through 8 full segments, through the molecular machinery of synapses and the sweeping systems of the whole brain, through the physics of energy and the psychology of false memories, through the map-making of place cells and the purposeful architecture of forgetting. It has been a long and winding path through some of the most intricate territory in all of science. And tonight we go deeper still. Into the territory that sits just below what most popular accounts of memory ever reach. The engineering beneath the biology. The precise, almost mathematical elegance of how the brain solves problems that human engineers are only beginning to understand how to formulate. This segment is, in some ways, the most technical we have visited. But we will move through it the same way we have moved through everything else, slowly, with long pauses, with the understanding that you do not need to grip any of this tightly. These ideas are not meant to be memorized. They are meant to wash over you, the way water washes over stone, leaving something behind that you cannot quite name but that subtly changes the shape of things. So let yourself be still. Let your breathing be slow and regular. And let's go a little deeper. The storage capacity of the brain's memory system is directly tied to the physical sizes of its synapses. Larger synapses encode stronger connections, and the range of possible sizes determines how much information a single synapse can store. We have spoken, many times now, about synaptic strength, the idea that memories are encoded in the relative strength of connections between neurons, with strong connections representing well-established associations and weak ones representing more tenuous links. But we have not yet asked a more precise question: how much information can a single synapse actually hold? The answer depends on how many distinguishable states a synapse can occupy. If a synapse could only be either strong or weak, on or off, like a light switch, then each synapse would encode exactly one bit of information. But synapses are not binary. Their strength exists on a continuum, represented physically by the size of the synaptic connection and in particular by the size of the dendritic spine that hosts it. The range of possible synapse sizes is substantial. The largest synapses in the cortex can be many times the volume of the smallest. And each distinct size along that continuum represents a different level of connection strength, a different potential state that the synapse could occupy. The question of how many of these states are meaningfully distinguishable from one another, how many distinct levels of strength the brain can reliably maintain and differentiate, is a question about the precision of the system. And it turns out that the answer to this question has profound implications for the overall storage capacity of the brain. The more distinct states a synapse can reliably occupy, the more information each synapse can encode. And multiplied across the brain's vast number of synapses, even small increases in the number of distinguishable states translate into enormous increases in total storage capacity. The size of a synapse is not merely an anatomical detail. It is, in a very precise sense, a unit of stored information. Neuroscientists can estimate the brain's memory capacity in bits per synapse, and recent measurements suggest each synapse may store around 4.7 bits of information, giving the whole brain a storage capacity far larger than previously estimated. In 2016, researchers at the Salk Institute for Biological Studies published a measurement that significantly revised upward the estimated storage capacity of the human brain. Using extraordinarily detailed three-dimensional imaging of brain tissue, they measured the sizes of a large number of synapses with unprecedented precision, and from those measurements attempted to calculate how many distinguishable size states each synapse could reliably occupy. Their estimate, approximately 26 distinguishable size categories, which corresponds to roughly 4.7 bits of information per synapse. And when that number is multiplied across the approximately 100 trillion synapses in the human brain, the resulting storage capacity comes out somewhere in the range of a petabyte, a figure we mentioned all the way back in our first segment, but which can now be understood in its precise technical meaning. What made this finding particularly striking was the precision it implied. For a synapse to reliably encode 4.7 bits, to occupy one of 26 distinguishable states, the biological machinery of synaptic regulation must be considerably more precise than previously assumed. The synaptic weights are not rough, approximate, noisy variables. They are, in some meaningful sense, finely calibrated. The biological system is achieving, at the molecular level, a degree of precision that approaches what we would expect from carefully engineered information storage systems. This is one of those moments in neuroscience where the measurement arrives and simply stops you. The brain is not approximately impressive. It is precisely, measurably, calculably extraordinary. Individual synapses are not stable fixed structures, they exist in a state of constant microscopic fluctuation, flickering between different size states on timescales of minutes to hours. And yet, immediately after marveling at the precision of synaptic encoding, we must hold alongside it something that seems, at first, to contradict it. Individual synapses are not still. They are not fixed at a particular size and held there permanently, like a setting on a dial that has been locked in place. They are constantly, restlessly, microscopically fluctuating. On timescales of minutes to hours, individual synapses can shift in size, strengthening slightly, weakening slightly, undergoing small structural changes as part of the constant molecular turnover that characterizes all living biological structures. The proteins that make up the synapse are being continuously replaced. The actin filaments that give the dendritic spine its shape are being continuously remodeled. The receptors on the synaptic surface are being continuously added to and removed from the membrane. This perpetual microscopic motion means that a synapse is never quite the same from one moment to the next. And yet, across the population of synapses that collectively encode a memory, the overall pattern, the relative strengths of different connections, the landscape of the memory trace, remains stable and coherent. The individual elements are noisy and fluctuating, but the collective pattern they encode is robust. This is a deeply biological kind of stability. Not the static, frozen stability of a carved stone, but the dynamic, continuously renewed stability of a living system, like the constancy of a flame, which remains recognizably itself even as the molecules that constitute it are in constant, rapid motion. The memory flickers. And in the flickering, it persists. Synaptic size changes operate on different timescales depending on their scale. Small fluctuations happen rapidly, while large, lasting changes require the slower biological processes of structural remodeling. Not all synaptic changes are equivalent in their speed, their magnitude, or their durability. And the timescale on which a change occurs is closely tied to the scale of the change itself. Small fluctuations, the microscopic flickering we just described, happen quickly. They are driven by the relatively rapid processes of protein phosphorylation, receptor trafficking, and actin dynamics. These changes can occur within minutes, and they are correspondingly impermanent. They represent the finest grain of synaptic adjustment, the most sensitive and most volatile layer of the system's responsiveness to activity. Larger changes, the kind associated with robust long-term memory formation, require slower processes. Growing a dendritic spine significantly larger, or stabilizing a new spine that has been created, requires structural remodeling that takes hours. It requires the synthesis of new proteins, which, as we established in our early segments, takes time and metabolic investment. It requires the physical rearrangement of cytoskeletal components, the insertion of additional receptor proteins, the restructuring of the presynaptic terminal on the other side of the synaptic cleft. This creates a natural hierarchy of timescales in memory consolidation. The immediate response to experience, the rapid, small adjustments of early LTP, happens within minutes. The structural consolidation that makes those adjustments durable happens over hours. The systems level consolidation that transfers the memory to long-term cortical storage happens over days and weeks. And the slow, thin updating of old memories by new related experience continues, in the gentlest and most gradual way, throughout a lifetime. Memory is not one process happening at one speed. It is many processes, nested within one another, each operating on its own timescale, each serving a different function in the overall architecture of what it means to remember. Memory storage is partly noisy by design. A certain degree of randomness in synaptic transmission is not a flaw to be eliminated, but a feature that helps the system generalize and avoid overfitting to specific experiences. Here is something that runs against most of our intuitions about how a good memory system should work. We tend to assume that noise is bad, that random variability in the system is an imperfection, a source of error, something to be minimized in the service of accurate, reliable storage and retrieval. But the brain appears to have been designed, at least in part, around the strategic use of noise. Synaptic transmission is inherently probabilistic. When an electrical signal arrives at a synapse, the release of neurotransmitter into the synaptic cleft is not guaranteed. It happens with a certain probability, and that probability varies with the strength of the synapse and the recent history of activity. There is a fundamental randomness built into the basic unit of neural communication. This noise, it turns out, serves important computational functions. In the encoding of memories, a certain degree of variability in how patterns are represented prevents the system from encoding experiences with such rigid specificity that only an exact repetition of the original cue can trigger retrieval. The noise introduces a kind of flexibility, a blurring of the edges of the encoded pattern, that allows the memory to be retrieved by approximate, partial, or slightly degraded versions of the original cue. It allows generalization. It allows the system to recognize that this is similar enough to that to be treated as the same thing. A perfectly noiseless memory system would be extraordinarily precise in its encoding but brittle in its retrieval, unable to recognize anything that didn't exactly match the stored template. The biological brain, with its designed-in stochasticity, trades some precision for robustness. And in doing so, it achieves something that engineered systems have struggled to replicate, a memory that works gracefully in the messy, approximate, endlessly variable world that biological creatures actually inhabit. Inhibitory neurons play a critical role in sculpting memory patterns. By suppressing the activity of some neurons while others fire, they sharpen the boundaries of encoded representations and prevent the spreading of activation that would blur memories together. We have spent most of our time thinking about the excitatory neurons, the cells that fire signals forward, that activate one another, that form the positive connections that constitute memory traces. But the brain contains a very large population of a different kind of cell, inhibitory interneurons, whose function is precisely the opposite. Rather than activating their neighbors, they suppress them. And this suppression is not merely a break, a way of calming the system down when things get too excited. It is an active, precise, computationally essential part of how memory works. When a memory is being encoded or retrieved, the relevant engram cells need to fire. But the success of the encoding depends not only on those cells firing, but on the surrounding cells remaining relatively quiet. The inhibitory interneurons enforce this quiet. They suppress the activity of neurons that are not part of the current memory pattern, sharpening the boundaries of the active representation, ensuring that the signal of this memory is cleanly separated from the background noise of everything else. Without this inhibitory sculpting, activation would spread, one memory would bleed into adjacent patterns, representations would merge, the distinctiveness of individual memories would be lost in a wash of undifferentiated activity. The inhibitory neurons are the contrast agents of memory. They don't create the image, but they sharpen it, making the signal clear and the boundaries precise. The inhibitory neurons are, in a very real sense, as responsible for what you remember as the excitatory neurons that do the remembering. Memory is the shape of the silence as much as it is the pattern of the firing. Memory storage is regulated by brain state switches, global neuromodulatory signals that shift the brain between modes optimized for encoding, consolidation, and retrieval respectively. Your brain does not operate in one fixed mode. It cycles through different global states, different configurations of neuromodulatory tone that shift the whole system toward different functional priorities. And these global state shifts have profound effects on every stage of the memory process. The major neuromodulators, acetylcholine, norepinephrine, dopamine, serotonin, are released broadly across large areas of the brain by small clusters of neurons in the brainstem and basal forebrain. When their concentrations shift, they change the operating mode of the networks they bathe, altering how readily synapses undergo plasticity, how strongly new inputs are encoded relative to stored patterns, how the hippocampus and cortex communicate with one another. High acetylcholine levels, for example, are associated with states of focused wakefulness and attention, states in which new information is strongly encoded and the influence of stored patterns is relatively reduced. This is the encoding mode. The brain is set to take in, to register, to write. Lower acetylcholine and different neuromodulatory balances, as occur during sleep and particularly during slow-wave sleep, shift the system toward consolidation mode, where stored patterns are replayed and transferred, and the influence of new inputs is reduced. The brain is set to process, to organize, to stabilize. And retrieval draws on yet another configuration, one in which stored patterns are most easily reactivated by partial cues, where the balance tips toward pattern completion rather than new encoding. The brain switches between these modes as naturally and as automatically as it switches between waking and sleeping. And each switch is, in its own way, a different relationship with memory. A different aspect of the full cycle of remembering, brought to the foreground for its moment, then giving way to the next. Cholesterol, produced locally in the brain and tightly regulated, plays an essential and often overlooked role in the formation and maintenance of the synaptic structures that support memory. Here is something that may feel unexpected in the company of everything else we have discussed tonight. Cholesterol. It is a word most commonly associated with diet and cardiovascular health, with something to be monitored, managed, reduced. Not something we typically associate with the elegant architecture of memory. But in the brain, cholesterol is essential. The brain produces its own cholesterol locally. It cannot use cholesterol from the bloodstream, which is prevented from crossing the blood-brain barrier, the protective boundary we will discuss in our next fact. The brain synthesizes its own supply, and it uses it in very specific and critical ways. Cholesterol is a major component of myelin, the insulating sheath that wraps around the axons of neurons and dramatically increases the speed of signal transmission. It is a crucial component of cell membranes, affecting their fluidity and the function of the protein channels embedded within them. And it is particularly important at synapses, where the membranes of the pre- and postsynaptic cells must be precisely structured to support the molecular machinery of synaptic transmission and plasticity. Research has shown that disrupting cholesterol metabolism in the brain impairs synaptic plasticity and memory formation. Astrocytes, the star-shaped support cells of the brain, which outnumber neurons significantly and perform a vast range of maintenance functions, appear to play a key role in supplying neurons with cholesterol, essentially manufacturing and delivering this essential structural material to the synapses that need it. Memory, it turns out, is partly made of cholesterol. The elegant biological machine of the mind runs, in part, on the same lipid that we monitor in our blood. The brain is found a use for everything. It wastes nothing. The brain guards its memory-supporting environment with a dedicated protective barrier, the blood-brain barrier, which tightly controls what substances can enter the brain and maintains the precise chemical conditions that memory processes require. The brain is extraordinarily sensitive to its chemical environment. The processes we have been describing throughout this series, the delicate dance of ion concentrations, the precise molecular cascades of LTP, the calibrated release of neuromodulators, the careful regulation of synaptic cholesterol, all of these require a chemical environment that is maintained within very tight parameters. Even small disruptions in the ionic balance of the fluid surrounding neurons, or the introduction of substances that interfere with synaptic signaling, can have profound effects on memory and cognition. The blood-brain barrier is the structure that protects this environment. It is formed by specialized cells that line the walls of the blood vessels within the brain, cells that are tightly joined to one another, with none of the gaps through which molecules can freely pass in other parts of the body. The result is a selective filter of extraordinary discrimination. Water, oxygen, and glucose pass through readily. Certain small molecules that the brain needs can cross. But large proteins, many pathogens, and most drugs are excluded unless they have specific chemical properties that allow them to cross. The cholesterol the brain requires must be synthesized internally, because the barrier excludes it from the bloodstream. Many medications that work elsewhere in the body cannot act on the brain because they cannot penetrate the barrier. The chemical world inside the blood-brain barrier is, in a very real sense, a different environment from the rest of the body, more precisely controlled, more carefully guarded, more specifically optimized for the extraordinary processes that happen within it. The brain does not merely perform memory. It maintains, with dedicated biological infrastructure, the precise conditions under which memory is possible. The memory system as a whole is optimized for energy economy. Every architectural feature we have discussed, from sparse coding to inhibitory sculpting to purposeful forgetting, reflects the pressure to do more with less. And so we arrive at the principle that ties together everything we have explored tonight, and much of what we have explored across this entire journey. The brain's memory system is not designed for maximum power. It is designed for maximum efficiency. For achieving the greatest possible computational performance within the tightest possible metabolic constraints. Sparse coding, the strategy of encoding memories and patterns where only a small fraction of neurons are active at any one time, minimizes the metabolic cost of any individual memory while allowing the combination space to be astronomically large. The fewer neurons that need to fire to represent a memory, the cheaper that memory is to encode, store, and retrieve. Inhibitory sculpting, the sharpening of memory representations by suppression of surrounding activity, reduces the energy wasted on irrelevant firing and keeps the signal clean without requiring additional hardware. Purposeful forgetting, the active removal of low-priority traces, frees up synaptic resources for new encoding and prevents the saturation that would make the system increasingly expensive to run. The blood-brain barrier, the maintenance of a precisely controlled internal environment, reduces the variance in operating conditions that the molecular machinery of memory would otherwise have to cope with, allowing it to be optimized for a narrower range of conditions rather than being built for robustness across a wide, unpredictable range. Even the stochastic noise of synaptic transmission, which we might initially have seen as a flaw, an energy cost that produces errors, turns out to serve functions of generalization and robustness that would otherwise require expensive additional mechanisms. Every feature of the memory system, when examined closely, reveals the pressure of the same fundamental constraint, the biological mandate to accomplish as much as possible at a cost that a living creature running on food can actually sustain. 20 watts. 1.4 kilograms. The accumulated wisdom of millions of years of evolutionary pressure. And the result is something that stores a petabyte of information in a self-correcting, self-maintaining, continuously updated, energy-efficient, noise-tolerant, generalization-capable, emotionally sensitive, spatially organized, temporally tagged, socially embedded, beautifully imperfect system that we call, simply, memory. We have reached the end of our 9th segment. And this one has taken us into the deep engineering of the brain, into the precise measurement of bits per synapse, into the flickering dynamics of individual synaptic connections, into the role of inhibition and neuromodulation and cholesterol and the blood-brain barrier and the fundamental design principle of efficiency that runs through all of it. What emerges from all of this, if you hold it lightly, is not a technical picture but an aesthetic one. The brain is beautiful. Not in a vague, general sense, but in a specific, precise, architectural sense, the way a well-designed bridge is beautiful, or a mathematical proof is beautiful. It solves hard problems with elegant solutions. It achieves extraordinary results within extraordinary constraints. And it does all of this without any of us having to think about it, or manage it, or understand it, or do anything at all. It does it while you sleep, which is exactly what it's doing right now. Let it. Good night. Welcome back to the quiet. We have arrived, together, at the final segment of this journey. The 10th. And if you have been here since the very beginning, since we first asked what it means that the brain can hold what amounts to hundreds of years of continuous experience, since we first descended into the world of synapses and engram cells and calcium ions and place cells and the purposeful architecture of forgetting, then you have traveled a very long way through some of the most intricate and beautiful territory that science has mapped. And if this is where you are arriving for the first time, drifting in somewhere near the end, catching the last hour of a longer conversation, that's fine too. Memory, as we have learned, is not linear. It is associative, contextual, distributed. It finds its way to what it needs from wherever it happens to be. Tonight, we conclude. And we conclude, as all long journeys should, by looking both back and forward. By taking stock of what we have learned about the rules that govern memory, and then by asking how those rules change across a life. How they are different in childhood than in old age. How early experience shapes the very architecture of the system that will carry all future experience. How memory, in a strange and profound sense, extends beyond the brain itself. And then, at the very end, we are going to let the science fall gently away. And we are going to sit, just for a little while, with the simple and remarkable fact of what it means to be a creature that remembers it all. Let's begin slowly. The way all endings should begin. Memory storage involves a fundamental trade-off between speed and stability. Fast encoding produces fragile traces, while stable long-term storage requires slow, resource-intensive consolidation. Throughout this journey, we have circled around a tension at the heart of the memory system without ever naming it directly. Tonight, in our final segment, we name it. The tension between speed and stability. Between the brain's need to encode new information quickly, to capture the present moment before it passes, and its need to store that information in a form that is durable enough to last. These two requirements pull in opposite directions. Fast encoding, by its nature, produces fragile traces. The rapid molecular changes that constitute early LTP can be reversed by subsequent activity, disrupted by interference, washed out by the normal molecular turnover of the synapse. Speed is bought at the cost of durability. Slow, stable encoding, the full protein synthesis-dependent consolidation process, the structural remodeling of dendritic spines, the system's level transfer from hippocampus to cortex, produces durable traces. But it takes time. Hours, days, weeks. It cannot happen at the speed of experience. Life does not pause to allow full consolidation before the next moment arrives. The brain's solution to this tension is the two-stage architecture we have described at length: encode quickly and temporarily in the hippocampus, then consolidate more slowly and durably in the cortex, primarily during sleep. It is an elegant engineering compromise. A staging area for fast capture, and a long-term archive for stable storage. Each optimized for its own timescale, and together solving a problem that neither could solve alone. Every memory you have ever formed was, at some moment, a fragile thing. Something that could have been lost. The fact that it survived, that it made it through the consolidation process and into something durable, is, in a small and quiet way, a kind of miracle. Everything you remember was, once, at risk of being forgotten. And something in the system decided to keep it. Some synapses act as meta-memory switches. Rather than encoding specific memories themselves, they regulate the threshold at which other synapses can undergo plasticity, setting the conditions for learning across entire circuits. And now, one final technical marvel before we turn toward the longer arc of a life. Because there is a class of synaptic modification that we have not yet discussed, and it is one of the most conceptually rich findings in the cellular biology of memory. Some synapses are not primarily engaged in storing specific memories. Instead, they regulate the plasticity of other synapses. They set the threshold at which neighboring synaptic connections can be modified by experience. These are sometimes called metaplastic synapses, or memory switches at the meta-level. They don't hold the content of memory. They determine the conditions under which memory can form at all. The concept is sometimes described as the plasticity of plasticity, the brain's ability to regulate its own capacity to change. A synapse that has been recently active may be made temporarily less able to undergo further LTP, a protective mechanism that prevents individual synapses from being perpetually saturated at maximum strength. Conversely, synapses that have been inactive for a period may have their plasticity threshold lowered, made more susceptible to change, readier to encode new information when it arrives. This metaregulation means that the brain is not just responding to experience. It is continuously calibrating its own responsiveness to experience. It is managing, at the synaptic level, how ready it is to learn, how open the system is, at any given moment, to being changed by what it encounters. The brain, in other words, has memory about its own memory. It tracks not only what has been learned, but how much learning has recently occurred, and adjusts its receptivity accordingly. It is, even at the cellular level, managing its own relationship with experience. The rules governing memory storage are not uniform across brain regions. Different areas use subtly different plasticity mechanisms, with different induction thresholds, different timescales, and different susceptibility to disruption. We have spoken throughout this journey about the diversity of memory systems, different circuits for different types of memory, different pathways for emotional versus declarative versus procedural storage. And this diversity extends all the way down to the rules of plasticity themselves. The cortex does not follow identical rules to the hippocampus. The amygdala uses different induction mechanisms than the cerebellum. The prefrontal cortex, so central to working memory and the organization of long-term autobiographical memory, has distinct plasticity properties that reflect its different role in the memory architecture. Even within a single brain region, different layers of cells, different subtypes of neurons, different subcircuits can have measurably different plasticity thresholds and timescales. The diversity is fractal. It repeats at every scale, from the whole brain level down to the individual synapse. This means that there is no single universal rule for how the brain stores information. There is a family of related rules, implemented in overlapping molecular machinery, tuned by evolution and development to the specific computational demands of each circuit. The hippocampus encodes fast and transfers slow, optimized for episodic memory. The cerebellum encodes through a different form of plasticity, optimized for the fine-grained motor learning that allows you to balance, to catch, to play an instrument. The amygdala encodes with extraordinary speed and emotional priority, optimized for the survival-relevant learning that cannot afford to wait for deliberate processing. The brain is not one memory system with one set of rules. It is an ecology of memory systems, each with its own rules, coexisting and interacting and together producing the full, rich, multidimensional experience of what it means to remember. The rules of memory storage change dramatically across development. Infancy, childhood, adolescence, and adulthood each have distinct plasticity profiles that shape what can be learned and how durably it is stored. And now we turn to time. Not the time of a single experience, or a single night of sleep, but the long time of a life. Because the memory system does not remain constant across a lifetime. It develops. It changes. It has different properties in different seasons of life. And those properties shape not just how we remember, but what kinds of learning are possible, and how deeply the traces of experience are inscribed. In infancy and very early childhood, the brain is in a state of extraordinary plasticity, far more so than at any later point in life. Synaptic connections are being formed at a breathtaking rate. The brain is, in a sense, building itself through experience, using the input it receives to wire its own circuitry, to establish the basic patterns of sensory processing and motor control and social responsiveness that will form the substrate for all future development. But this extraordinary early plasticity has an interesting limitation: the hippocampus, and particularly its capacity for the kind of explicit episodic memory that we can consciously recall and narrate, is not yet fully mature. This is widely thought to be a significant contributor to the phenomenon known as infantile amnesia, the near-universal absence of autobiographical memories from the first 2 to 3 years of life. Not because nothing was being encoded, but because the hippocampal system that supports explicit, consciously recallable episodic memory was not yet operating in the way it will in later childhood and adult life. What was being encoded, and powerfully, was everything else: the emotional patterns, the sensory associations, the procedural and implicit learning that forms the deep substrate of who we become. The explicit memory is absent from those early years. But the implicit memory is very much present, shaping personality and attachment and emotional response in ways that can persist for an entire lifetime. Memory storage is tuned across the lifespan, with distinct sensitive periods in childhood where specific types of learning are maximally efficient, followed by gradual changes in plasticity that continue into old age. Beyond the earliest years, development continues to reshape the memory system in ways that are less dramatic but no less significant. The concept of sensitive periods, windows of development during which specific types of learning are maximally efficient, has been established across many domains. Language acquisition is perhaps the most familiar example. Children learn language with an ease and a completeness that adults cannot match, not because they are more intelligent, but because the neural circuits for language acquisition are in a state of heightened plasticity during early childhood that gradually diminishes with maturation. The same language that a 6-year-old acquires effortlessly, a 40-year-old must learn through deliberate effort and with an accent that the child would never develop. Musical training, certain aspects of visual processing, the acquisition of social and emotional skills, all of these have sensitive periods. Windows during which the relevant circuits are maximally receptive to the experience that shapes them. After these windows close, after the circuits mature and their plasticity settles into adult configurations, learning is still possible, but it is harder, slower, and less complete. Adolescence brings another wave of profound neural reorganization, the extensive synaptic pruning that we mentioned in our discussion of active forgetting, the dramatic remodeling of prefrontal circuits that underlies the development of executive function and impulse control, and simultaneously a period of heightened sensitivity to social experience and emotional learning that makes adolescent memories often among the most vivid and formative of a lifetime. In old age, plasticity changes again, slower, perhaps more difficult to trigger, but not absent. The aging brain retains the capacity to form new memories and to learn new things. The rules have changed. The efficiency has declined. But the fundamental capacity, the biological potential for experience to leave a trace, endures. Early life experiences can permanently reshape the brain's memory storage rules, altering the plasticity thresholds, the stress hormone response, and the emotional architecture of the memory system in ways that persist across a lifetime. This is perhaps the most consequential fact in this entire series of segments, and it deserves to be held with care. The experiences of early life, particularly the quality of caregiving, the presence or absence of safety and predictability, the emotional tone of the earliest relationships, do not merely create memories in the ordinary sense. They shape the very machinery that will form all future memories. They alter the plasticity thresholds of the developing brain, the calibration of the stress hormone response system, the sensitivity of the amygdala, the capacity of the prefrontal cortex to regulate emotion. A child who grows up in an environment of consistent, responsive care develops a stress response system that is calibrated for a world in which challenges are manageable and support is available. A child who grows up in an environment of chronic unpredictability or threat develops a stress response system calibrated for a more dangerous world, more reactive, more sensitive to threat, more readily flooded by the cortisol that, in high and sustained doses, impairs hippocampal function and distorts memory encoding. These calibrations are not destiny. The brain retains plasticity throughout life, and with the right experiences, relationships, and sometimes therapeutic support, the calibrations of early life can be shifted. The rules can be partially rewritten. But they are real. They are durable. And they represent one of the most important truths that the neuroscience of memory has to offer about the conditions under which human beings flourish. The earliest experiences are not merely the first entries in the memory archive. They are the opening instructions that shape how the archive itself is organized, what it can hold, and how it responds to everything that comes after. The care we offer to the very young is not merely kindness. It is neuroscience. Task-specific circuits have their own memory storage rules. The neural machinery that remembers how to ride a bicycle uses fundamentally different plasticity mechanisms from the machinery that remembers where you were when you learned. We have returned, one final time, to the theme of diversity. To the reminder that memory is not one thing but many, not one system but an ecology, not one rule but a family of rules, each adapted to the specific demands of the circuit that uses it. The cerebellum, which is essential for motor learning and the fine-grained calibration of movement, uses a form of plasticity called long-term depression, a weakening of synaptic connections rather than a strengthening, as its primary mechanism for learning. This is the opposite of the LTP that dominates our discussions of hippocampal and cortical memory. And it produces a different kind of trace, precise, automatic, deeply embedded in the motor machinery, expressed not in the currency of conscious recollection but in the fluid, effortless execution of practiced movement. The striatum, involved in habit formation and the learning of stimulus-response associations, uses yet another set of plasticity rules, ones that operate on different neurotransmitter systems, respond to different teaching signals, and produce a type of memory that is rigid and automatic in a different way than either episodic memory or motor memory. Each of these systems is doing something genuinely different with experience. Each is taking a different slice of what happens to you and transforming it into a different kind of trace, a different kind of influence on future behavior. And together, they allow a single human being to simultaneously remember facts, recognize faces, ride bicycles, develop habits, feel familiar emotions in familiar contexts, and navigate through the world with the accumulated benefit of everything that has come before. You are not one kind of memory. You are all of them, simultaneously, continuously, for as long as you live. Memory is not confined to the brain. Other organs, particularly the heart and gut, contain neural tissue and have forms of cellular memory that can influence behavior and well-being in ways science is only beginning to understand. And now, one of the most quietly astonishing facts in this entire series. Because memory, it turns out, does not stop at the skull. The gut contains an extensive network of neurons, sometimes called the enteric nervous system, sometimes described as the second brain, comprising hundreds of millions of nerve cells that manage the complex processes of digestion and gut function, but that also communicate bidirectionally with the brain via the vagus nerve. This gut-brain axis is involved in mood, stress response, and behavior in ways that are only beginning to be understood. And there is growing evidence that the enteric nervous system has forms of plasticity, forms of cellular change in response to experience, that constitute a kind of peripheral memory. The heart, too, contains its own intrinsic nervous system, a network of neurons that can act semi-independently of the central nervous system, and that maintains its own records of cardiac experience, its own learned patterns of response. The immune system has memory, the immunological memory that allows it to respond faster and more effectively to pathogens it has encountered before, a form of cellular learning that is quite distinct from neuroplasticity but that serves an analogous function. And perhaps most profoundly, every cell in the body carries epigenetic marks, the chemical modifications to DNA packaging that we discussed in our early segments, that reflect the history of the organism's experience, that carry, in a biochemical form, a record of what has been lived. Every cell remembers, in its own molecular way, something of what the whole has been through. Memory is not a brain event with bodily consequences. It is a whole organism phenomenon. The brain is the most elaborate and flexible memory system in the body. But it is not the only one. You remember with your whole self. More literally than you knew. A specific molecular switch, a kinase called pKm-zeta, or related molecules in its family, appears to be essential for maintaining long-term memories, and disrupting it can erase memories that have been stored for months or even years. Here is a fact that sits at the intersection of the remarkable and the sobering. We have spoken of memory as durable, as something that, once consolidated, is built into the physical structure of the brain in ways that are resistant to disruption. And in many ways, that is true. Long-term memories are robust. They survive sleep and distraction and the passage of time. They persist through the continuous molecular turnover of the proteins that constitute them. But there is a molecular switch that appears to be essential for their maintenance. A kinase, an enzyme that modifies other proteins, that must remain active at strengthened synapses in order for the memory to persist. When this switch is pharmacologically blocked, even in animals with well-consolidated, months-old memories, those memories can be erased. Not weakened, erased. The behavioral evidence of the memory disappears, apparently completely. This finding, first clearly demonstrated in the early 2000s and subsequently extended and refined by many research groups, suggests that long-term memory is not merely a static structural trace, a fixed pattern of synaptic weights that once established requires no further maintenance. It is, at least in part, an actively maintained dynamic state. A state that depends on the continued activity of specific molecular switches that must be continuously reset and reinforced. The implications are profound in multiple directions. They raise the possibility, deeply uncomfortable and ethically complex, of pharmacological memory erasure, of deliberately disrupting specific memories in clinical contexts, for people suffering from the intrusive, disabling effects of traumatic memory. And they deepen our understanding of memory as a process rather than a product, something that is not merely stored, but continuously, invisibly, sustained. Memory is not a monument. It is a vigil. And something in the brain keeps watch. The brain compresses memories over time. Recent memories are stored in rich, detailed, episodic form, while older memories are gradually distilled into more compressed, schematic representations that capture the gist while releasing the specifics. And here, at the very end of our journey, we arrive at something that feels, in the most fitting possible way, like a completion. Like the form of the thing revealing its meaning only when you can see the whole of it. Memory is not merely preserved over time. It is transformed. The rich, detailed, sensory, emotionally vivid episodic memory of a recent experience, the full resolution version, with its particular light and its specific words and its precise emotional texture, gradually, over months and years, gives way to something more compressed, more schematic. A kind of essence of the experience, rather than the experience itself. The specific words of a conversation fade, but the feeling of it remains. The precise sequence of events on a particular day becomes indistinct, but the overall character of that period of your life, what it was like to be you then, in that place, at that time, is distilled and preserved in a form that is less detailed but often, strangely, more meaningful. The gist survives what the specifics cannot. This compression is not only loss. It is also a kind of wisdom. The brain, over time, extracts from the flood of particular experience the patterns and essences and emotional truths that are most worth carrying. It lets the pixels go and keeps the painting. It releases the exact words and keeps the meaning. It distills, gently and continuously, across the long span of a life, the most essential version of what has been lived. And so the record that an old person carries, the memory of a life fully lived, is not a high-resolution archive of every moment. It is something more like a great work of compression. A distillation. A collection of essences, each one the product of years of the brain's patient curatorial work. Not the raw footage. Something closer to art. We have reached the end. The 100th fact. The final breath of a very long journey. And what has this journey given us? Not a complete account of memory, no account can be complete, and the science continues, daily, to discover new depths and new surprises in the biology of remembering. What it has given us, perhaps, is a new kind of intimacy with the process. A sense of what is actually happening, physically and chemically and electrically, in the silent activity of the brain that carries your life. You contain, in the tissue of your brain, the molecular traces of every significant thing that has ever happened to you. Every person who has mattered. Every place that has left its mark. Every moment of fear or joy or wonder or grief that was significant enough to earn the investment of consolidation. All of it is there, in the pattern of synaptic weights, in the configuration of engram cells, in the epigenetic marks on your DNA, in the spatial maps of your hippocampus, in the compressed and distilled essences of experiences that the brain has been quietly curating for as long as you have been alive. You are not separate from your memory. You are, in the deepest sense, made of it. The person you are, the particular texture of your attention, the specific geography of your emotional responses, the things that frighten you and the things that move you and the things that make you feel, however briefly, completely at home in the world, all of it has been shaped, continuously and cumulatively, by the record that your brain has been keeping since before you were old enough to know that a record was being kept. Memory is not something you have. It is something you are. And the brain that holds all of this, your particular brain, with its particular history, its particular patterns of synaptic weight, its particular map of everywhere you have ever been, is, right now, in the quiet dark of wherever you are, doing what it has always done. Consolidating. Replaying. Distilling. Keeping watch. You don't need to help. You never did. You just needed to live. And remember. And occasionally, on a quiet night like this one, to let someone remind you of the extraordinary thing that has been happening inside you all along. Sleep well. And thank you for listening.